Cuando nosotros vemos a Latinoamérica, el Caribe y España, no vemos las fronteras, vemos las necesidades, las oportunidades y también las esperanzas compartidas por más de 17 millones de agricultores familiares. Ellos producen la mayor parte de los alimentos de la región que contribuyen a la sostenibilidad ambiental y la biodiversidad, pero también enfrentan los desafíos de sostener y mejorar sus sistemas productivos en un contexto de crisis climática, a través de la tecnología y la innovación aplicadas a los sistemas agroalimentarios. Desafíos sin fronteras que requieren soluciones sin fronteras. Somos FONTAGRO, una plataforma de cooperación internacional para el desarrollo y cofinanciamiento de proyectos de ciencia e innovación agropecuaria, fundado hace más de 26 años, para impulsar la investigación e innovación en la agricultura regional, reconociendo su importancia prioritaria. En América Latina y el Caribe, contamos con una sólida estructura de gobernanza conformado por un consejo directivo, una secretaría técnica, administrativa, para unir esfuerzos y atraer inversiones públicas y privadas en el sector agropecuario, formada por una alianza de 15 países miembros, con el respaldo del BID, el ICA, Ministry for Primary Industries, el Global Research Alliance, el Global Methane Hub, FAO, CGIAR y Alianza Bioversity SIAT. Impulsamos proyectos colaborativos entre los investigadores de los Institutos Nacionales de Tecnología de los 15 países de América Latina, el Caribe y España, con quienes hemos contribuido a la realización de 204 proyectos de innovación, con una inversión de más de 150 millones de dólares. En la actualidad estamos ejecutando 59 proyectos que conectan los esfuerzos de 1.500 investigadores en campos tan diversos como cultivos, ganadería, alimentos y mercados, recursos naturales, producción forestal y agricultura. Somos FONTAGRO, ciencia colaborativa en acción para un futuro sostenible e inteligente.
Good morning and welcome. This is a great honor to open this FONTAGRO webinar. One of the most urgent animal health challenges of our time is the re-emergence of the new World Screwworm. This pest is not just a veterinary concern, it directly threatens the health of our cattle and livestock, undermines beef and dairy production, and endangers the livelihood of farmers across our region and the hemisphere. The larvae infest open wounds, causing severe pains, decreased productivity in many cases, and the loss of animals. For producers, especially the small and the medium farmers, these losses translate into very important reducing incomes. The economic stakes are enormous. In past outbreaks, annual cost had reached very important amount. And what makes this challenge especially complex is the transboundary nature. The screwworm does not recognize borders. An infestation left uncontrolled in one country quickly becomes a risk in other countries. And this is why coordinated hemispheric actions is not optional, is really essential. This is where collective responsibility comes in. Farmers are the first line of defense. They monitoring their cattle and they try to report the cases they found. National animal health offices must strengthen surveillance, implement effective eradication strategies, and coordinate across borders. And science, our research and academic institutions must continue to innovate, whether through improved diagnostic or sterile insect techniques or management practices or all a combination of all the others. But above all, success depends on working together. No single institution or government can solve this problem alone. We need cooperation across countries, across disciplines, across institutions, especially university and research institutions. And FONTAGRO role is precisely to create that bridge, is bring farmers associations, academic institutions, research institutions, and also different government agencies in one shared conversations. And today's webinar is a step forward on that. It's an opportunity to learn from experts, and thank you very much to all of you that are in this webinar. And thank you very much to all the people that is also online. And thank you, and special thank you for all the experts that we have in the webinar, from ICA, Jose Ordaz, people from the University of Florida, Texas A&M, the INEA Uruguay, Foundation for Food and Agricultural Research, and again, all the people online. So now I want to welcome Andrew Short. Andrew, the floor is yours, and thank you so much for joining us.
Thank you so much. Thank you for organizing this. And my name is Andrew Short. I'm the chair of the Department of Entomology and Nematology at the University of Florida. I'm also the vice president-elect for the Entomological Society of America. When this outbreak emerged, we recognized that for much of Latin America, like it never went away. This has been something that has been dealt with and continues to be a problem for a long time. But when this outbreak emerged and kind of threatened Central America and the U.S., our University of Florida, Texas A&M, and Oklahoma State University kind of got together, since we're in states that are significantly at risk for economic damage to this pest. And we represent some of the largest entomology departments in the country, both in research and training programs. And so we wanted to make sure, since we all have capacity in veterinary entomology and have some level of experience fighting New World Screwworm and doing research in this space, that we were able to contribute to finding a solution to combating this issue, not only for the US, but also throughout the rest of Latin America. And so I think that's why we wanted to make sure we could get our experts together with all the other experts together and really put together our ideas to come up with some identify gaps and the best solutions for this really critical problem. So thank you all for coming, and we're happy to contribute whatever we can to help find solutions. And with that, I am going to turn it over to Bola Adesogun.
Good morning, everyone. I hope you can hear me. Thank you so much for joining us, colleagues, partners. I am Bola Adesogun. I'm Associate VP and Director of the Global Food Systems Institute and E.T. York Professor of Agriculture. My background is livestock nutrition, and it's really my honor, on behalf of Dr. Scott Angle, our Senior VP of Agriculture and Natural Resources, who leads IFAS, our Institute of Food and Agricultural Sciences at the university, to welcome you to this workshop. We're here to address the urgent and complex threat of the New World Screwworm. And now, with the first human case, it's uppermost on the minds of a lot of people here in the US. We're deeply grateful to FONTAGRO for co-hosting this meeting with us, and to all of you, representatives from leading universities, generous donors, dedicated experts who share a commitment to safeguarding animal health, food systems, human health, and supporting livelihoods. Here at UF, we are proud to stand at the intersection of science, innovation, and global collaboration. We have long recognized that challenges such as this, just like Eugenia mentioned, cannot be solved in isolation. They demand collective vision, coordinated research, and sustained partnerships across disciplines, sectors, and borders. We're very privileged to have Dr. Andrew Short, chair of our Entomology and Nematology Department, leading the UF New World Screwworm effort, combat efforts on our behalf. He's spearheading everything, and we're really privileged to have him helping us with this effort. Once more, I want to thank each of you for joining us. We really look forward to a great discussion that will help us with strategizing, help us with jointly coming up with approaches that will effectively curtail this threat. So with that, once more, I'd like to thank FONTAGRO. I'd like to thank ICA, several of our donors here present, FFAR, USDA, our colleagues from leading universities. We're really honored to have you on the call, and we look forward to an exciting time of discussion. I will apologize. I'll be in and out because I just had surgery, so I'm recovering. So please bear with me. Well, thank you very much.
Jose?
Yes. Good morning, everyone. Once again, my name is Jose Urdaz. I'm the manager of the Agricultural Health, Food Safety, and Quality of Agri-Food Systems at AICA. I'm now in Costa Rica at the moment. So to me and everyone here, I believe this is truly an honor to welcome you to the opening of this very important workshop dedicated to tackling the threat of New World Screwworm. I would like to especially thank FONTAGRO and UF, the University of Florida, for leading this initiative, which also has allowed us to bring the vision and commitment of the Inter-American Institute for Cooperation on Agriculture, AICA. The screwworm, you know pretty well, knows no borders and no sectors. It affects cattle, domestic animals, wildlife, and people. Its reappearance in Central America and Mexico since 2023 is a strong reminder of a fundamental truth. Sanitary achievements can only be sustained through continuous vigilance, investment, and effective cooperation, as we are doing now here today. At AICA, we have assumed an active and committed role through a joint plan with APHIS USDA. We support countries with technical training and risk communication. We also respond to demands with our own internal resources, but our action goes beyond technical assistance. We work on behavior change, strengthening community networks, and connecting producers, veterinarians, animal health authorities, human health professionals, teachers, media, and civil society. We are in a moment that we've never seen this before for over 20 years, and now we need to relearn. We need to educate more. We need to, once again, get to know this disease. So now we know the sterile insect technique is powerful, but it cannot work alone. Eradication requires an integrated approach, particularly good livestock practices. Even today, as we speak, there's still many gaps in the way that farmers work with their animals that they need to improve in order to reduce this disease. Timely wound treatment, biosecurity measures, community education, and above all, shared responsibility. This workshop is a great opportunity to strengthen the link between science and international cooperation. I am very confident that the ideas and commitments arising here today will strengthen the roadmap toward the definitive eradication of the screwworm. It's a must. It's not that we can or not, it's a must. So with sustainability and shared responsibility. On behalf of AICA, I once again thank you, and I wish you a very productive workshop with open discussions, creative solutions, and a strong spirit of collaboration. Thank you very much.
Thank you, Jose. And Josh, I pass the floor to you.
Thank you very much, Eugenia. So during this meeting, we're going to have 3 panels. The first panel is surveillance, outbreak response, and risk modeling. The second panel, sterile insect technique and new biotechnologies. And the third panel, on-farm control measures and private sector involvement. So to moderate the first panel with Dr. Phil Kaufman and Dr. Lindsay Campbell, I will pass the floor to Dr. Andrew Short.
Thank you so much, Josh. And yeah, I'm happy to moderate this first panel with 2 speakers. This panel is kind of covering the surveillance, risk management, and outbreak response for New World Screwworm. And our first speaker, Dr. Phil Kaufman, who is chair and professor of entomology at Texas A&M, has been managing a lot of the screwworm efforts and extension efforts in the state of Texas, which is kind of on the front lines right now in the US for New World Screwworm. So I'm happy to pass it over to Dr. Kaufman.
Thank you, Andrew. Greatly appreciate being invited to this meeting and going to share what we know. As you're well aware, Texas is the most likely place for an established screwworm infestation to occur, given our proximity to where the cases are in southern Mexico now. And our livestock and wildlife associations have been very proactive in raising the awareness level and working with our state and federal partners to ensure that Texas is ready should the fly get this far north. So we were given a couple of charges for this meeting, and I'll share a PowerPoint here in a minute to share what we know. And I'm going to, just so they're aware of it, I'm going to defer some of the questions maybe to some of the others on this call who may be better suited to answer some of the, if we get into fine details. And just for reference, I was at the University of Florida before I moved to Texas A&M 5 years ago. And so I'm very familiar with the capabilities of both of these universities. And quite frankly, they're the top 2 universities in entomology, in my opinion, in the US. Let me put up the first PowerPoint. And so Dr. Campbell assembled this, and I'm going to present the first part of it and then turn it over to her for the second part. And we were tasked with best practices for case detection traps, GIS models, and genetic tools for tracing outbreaks, lab quality assurance, and quality control, as well as data standards. And it's kind of a big area, and a lot of what is in this request is not really in the purview of what we as universities are able to do. But I have very good connections with our USDA APHIS, who carries the responsibility for screwworm management in the US, as well as our USDA Agriculture Research Service, which is the research arm of the USDA in the United States. And we're fortunate to have the laboratory that leads the efforts on screwworm here in Texas, in Kerrville, which is about 3.5 hours from where I'm located in College Station. And it's the laboratory that Dr. Nippling worked at during his career. And he's the gentleman that came up with the sterile insect technique that we still rely on. So best practices for case detections, what we rely on and will continue to rely on is using trapping techniques that have been used over the years. And this has gained a renewed interest. So I have a couple of images up here that the USDA ARS laboratory, and I'll probably just refer to them as ARS going forward, laboratory has a postdoc, and she's pictured here. I believe it will be on your left side of your screen, hanging a screwworm trap. And one of the things just to bring attention to, and what this opportunity may present for us, is that research on screwworm in the United States really, for all intents and purposes, stopped once that fly was eradicated from the US. And that's because it's considered a foreign animal disease pest by our government, which means that we are not permitted to have that fly, live versions of that fly in the US. It would take extraordinary efforts that to bring that fly here. I'm hoping that with the announcement of our Secretary of Agriculture to build a new production plant in South Texas, that we can leverage that opportunity to begin doing research on the fly again, because quite frankly, that's what's hampered a lot of the research on this fly over the years, is the fear that if it's brought here that it would escape and we would be back where we were 50 years ago in this country. And so the ARS has a team of 3 really good scientists that rotate through the Pecora, Panama production plant at Copeg to conduct research. And unfortunately, what often happens is that if the research that they hope to conduct might interfere with the production facility, then that research is deemed not possible. So things such as investigating new insecticides, that type of work can't occur there, because the risk of that insecticide perhaps moving into their production colony is considered too great, and they don't want to do anything to disrupt the production of the sterile flies, which have traditionally been released over the Darien Gap to prevent what we're currently experiencing, where the flies get moved past the quarantine area. And so on the right is a PVC pipe, which is what USDA is currently using. And they essentially cover this pipe with a really adhesive material called Tanglefoot, which is very, very, very sticky and not nice to use. And around the side of that, you see a tube, and that tube is essentially filled with the attractant. And the attractant that has been used for decades is called swarm lure, and it's a mixture of 10 chemicals that has been shown to be the screwworm females in particular have been shown to be responsive to that chemical. The chemical is not easy to produce. The swarm lure is not easy to produce, but it is fairly effective. The problem with swarm lure is that it is not specific to and so it attracts essentially all types of blowflies, which is not helpful when you're trying to survey for screwworm in an area. Everything that's caught has to be looked at, and at least in the U.S., we have an additional fly in the same genera. Cochleomyia omnivorax is the screwworm, the New World Screwworm. We have what's called the secondary screwworm here, which is Cochleomyia macellaria, which the larvae are fairly easy to tell apart. The adults are much more difficult to tell apart. And so whenever we trap, you have to differentiate those flies as well as all of the other flies that will be collected. And so one of the things that USDA has recently advanced is they now have a product called Swarm Lure 5 that they have put out. And the only reason that they developed Swarm Lure 5 is that one of the chemicals that's a constituent of Swarm Lure 4 can no longer be shipped by airplane. And so they had to find a replacement product for that, and they did. But unfortunately, the Swarm Lure 5, which is the newest version, is slightly more attractive to the other blowflies than it is to the New World Screwworm. And so haven't really advanced in that area. And you'll see on one of the slides I'll have is opportunities that I think is part of the panel discussion is trying to work toward finding either a pheromone or an aggregation chemical that we can leverage to move forward in surveillance and trapping. And so what we're often left with is heavy reliance on particularly livestock producers reporting infestations of their animals. And that's really not a very good way to run a surveillance program, but it's kind of the best we have at this point. So what Texas is currently doing is our Texas Department of Agriculture is working with the USDA APHIS program, which is really responsible for screwworm, and they are monitoring areas. Right now, they're monitoring all along the southern border of the U.S., essentially from a city named Del Rio all the way down to the tip of Texas in Brownsville. And at first they had 100 traps that they were placing on the U.S. side of the border, and they were working with Mexican officials to get them to trap on their side of the border. But the current plan for Texas, and it's not quite released yet, so this is a bit of a speculation from what I have learned in my meetings, is that if a case is found in Texas, the immediate plan is to monitor areas surrounding a known infestation site to see if they can very quickly catch any screwworms that might be there, which tells them how long that population might have been in place. But once they start dropping sterile flies, which they tell us they'll do within 48 hours of a confirmed infestation, they will stop adult monitoring, because the traps are also very effective at catching our very expensive and very resource limited sterilized flies. The Texas plan is to create an immediate 6.5 kilometer radius buffer around a known case and immediately have what they call a stop animal movement. So no mammals will be allowed to move outside of that quarantine area until they fully establish their quarantine, which means posting signs and monitoring traffic and movement of livestock in particular on our highways surrounding this infestation area. And part of that is to make sure that we don't have inadvertent animals getting moved out, but also to maintain our current plan of regionalizing infestations if they occur in Texas, so that animal movement outside of those areas is not restricted. Texas is a very, very large state, and what we don't want is the entire state being quarantined by other states in the US, as well as our export markets, because of an interception of a fly. To give perspective, so Amarillo, Texas is up in our northwestern panhandle, and that's up in the area where all of our feedlots are located. The distance from the southern point in Texas or southern Texas to Amarillo is equivalent to the distance from that area of southern Texas to where the screwworm's currently infested. So it's about, I think, 800 kilometers distance. And so what we're trying to do is avoid a quarantine of the entire state, which would affect many of our livestock markets, and of course have economic damage when recognizing the great distances that occur here. So in an area where an infestation has been found, the plan is to have multiple weeks of sterile fly releases, both from ground-based and aerial distributions, before an area is considered cleared for normal operations. And so the plan is, if the fly is found in Texas, that a portion, if not all of the flies currently produced in Pecora at Copeg would be redirected up to Texas and then loaded into the airplanes for releases. There was one of the charges on this was looking at tracebacks. And so Texas has a very good plan for animal tracebacks already. So we have the cattle fever tick, we call it here, Rhipicephalus microplis. It used to be called Buophilus microplis. And that tick is in a quarantine area in very southern Texas. And we have infestations of it on some of our ranches in the state. And those ranches are essentially quarantined. And whenever a new case is identified, our Texas Animal Health Commission, which is equivalent to a Department of Agriculture in many other states, they automatically do what they call trace backs and trace outs. So where did any animals on that ranch originate from within a specified period of time, and where did any animal on that ranch or nearby ranches get moved to? And that's the plan to use here, is that if we do have an infestation, they're going to look back and they're going to look forward, so that we can start surveillance in areas where animals from that ranch or neighboring ranches may have been moved, to try and get ahead of an infestation rather than waiting for ranchers to report new cases. And this is where we get a bit outside of my expertise, but so when Florida had its outbreak of screwworm back in 2016, the big question was, where did those flies come from that originated that infestation? And at the time, the USDA scientists attempted to use some technologies, but they didn't have a very large pool of screwworm data from other countries and regions in South America and even the Caribbean, where the fly was known to exist. And so what they use now, and they've done a retrospective look back, they use single nucleotide polymorphisms, and it's kind of colloquially called DNA fingerprinting. So patterns of DNA that they look at in the genome of the screwworm, which has been sequenced, which is fantastic. And they compare that to whether those strings of DNA occur in other populations that they have data from. And using that, they work to determine where the likely source of flies were. But understand that that approach is not likely to work when we get an infestation in Texas, because the current infestation, it's been determined that the flies that made it past the quarantine all had originated from Colombia. And so the current infestation is like a lot of our invasive species. There's not a lot of genetic diversity. It probably was just a handful of organisms that created this current infestation. And so it's kind of looking at a family tree that doesn't branch very well. And so if we did tracebacks with SNPs on this, it likely isn't going to be, we wouldn't be able to say that population on ranch B came from a population on ranch A, because they're all related to each other. And there was a question regarding quality control, and I'm happy to elaborate if more direction can be provided during the panel, is that quality control in the co-peg rearing facility is all handled by USDA APHIS. And I'm not familiar with all of their metrics, but I do know that at every generation they evaluate sterility. They have production measures that they measure against, probably amount of pupae produced or eclosion of those pupae pre-sterilization and those that have been sterilized. And I know they regularly conduct mating studies. And the last part was on data standards, and I'm not sure how to address that. And I believe the other aspect was, and this may fall into panel 3, so apologize if I'm jumping ahead, is resources for extension. And I see it is in panel 3. So I believe I left this slide in that slide deck as well. If not, I'll move it back over there. But we do have a lot of resources that Texas A&M University has put forward since this outbreak. And I know the University of Florida has also worked on some of theirs. And I believe so we'll come back to these. I had sent these to Josh yesterday for research priorities and actions. Those that I put in red are those that I think are more relevant to this current panel, and we can bring these back up later as needed. And just coming back to the SNPs, from what I understand from USDA, is they have the capability of what they call the inner Caribbean, which is the yellow areas, the outer Caribbean, which is the kind of Dominican Republic and the rest of the Caribbean islands east of there, as a separate population, and then essentially all of South America. And using these regions is how they came up with that the flies originated out of Colombia from the current outbreak. But recognize that even with their current, as I understand their current process, they could always use more specimens for adding to their comparisons. A lot of the specimens they have are historic and not necessarily contemporary specimens. And then this is the phylogenetic dendrogram for relatedness. And so you can see there's a pretty big break between the South American populations and the inner Caribbean versus the outer Caribbean, which have a nice branch off of that. But they fall somewhere in between those two populations, which makes sense. And with that, I will turn it over to Dr. Campbell. And let me stop sharing.
Okay, thank you, Phil. So my name is Lindsay Campbell. I'm at the University of Florida. I'm at the Florida Medical Entomology Laboratory in Vero Beach, and I'm also in the Department of Entomology and Nematology. And so My research in lab focuses primarily on spatial modeling and geographic modeling and spatiotemporal distributions. So I'm going to give a little bit of an overview about some geospatial technologies and different, just barely touch on a couple of models that are useful in or potentially useful for New World Screwworm. So I'm just trying to make sure that I pull up the correct screen here. Okay, is this in the presenter view or can you still see notes? Okay, great. So everyone here is aware that New World Screwworm obviously has a very broad geographic distribution, particularly here in the Western Hemisphere. So thinking about geospatial tools such as maps, there's been a long history of including maps and trying to look at the distribution of this fly throughout different time periods of control and eradication. So this is a map from Bushland from 1985. And here you can see it's just kind of a hand drawing of what the general migration area might be in the southern United States or overwintering survival. And so these maps were used to go ahead and identify just these very general areas where there could be seasonal spread. You can see they've set up this eradication area in the southeastern portion of the US, and then moving into Mexico, where there could be some control and eradication activities. So if we fast forward to the current time period, there's been quite a few advances. So this is a recent map from USDA APHIS showing the distribution of this recent reestablishment across Central America. And this was from August 16th. And it all the way is showing, this recent outbreak in Veracruz in Mexico. So when thinking about, the past eradication or control efforts that were used, and then also the present technologies or present day situation, there's kind of, there's all these exciting new technologies, but there are also quite a few complexities that have made things potentially a little bit more challenging across these broad geographic areas. And so one of them is, ongoing livestock intensity has changed quite a bit over the previous eradication period. So this is just a map showing different densities of cattle and horses and pigs just across Central America and the southern US. But even though there's kind of these different changes in the landscape, fragmentation, a lot more movement of people and different activities, there's quite a few improved geospatial tools and technologies that can be used or potentially applied to this problem. And so that's what I'm going to be talking about today, is just touching on some of these different technologies, just a few basic examples of where they've been used to address this problem, and then linking this back hopefully to kind of this broader topic of transboundary integrated pest management and how these tools might be able to be used across the full distribution of New World Screwworm for improved communication, targeting of control, and other activities. So one thing that is advanced quite a bit is that it is possible now to have near real-time reporting of New World Screwworm outbreaks if people have access to internet or to phones that can go ahead and record geographic locations, then it's possible to communicate that information more quickly and rapidly to a central location like a dashboard for improved communication about the timing and the distribution of outbreaks that are taking place. There's also vast quantities of environmental data that's available globally, that's freely available and open source. And these include things from temperature and precipitation, humidity, and these are at multiple time scales. So they can be very fine from, hourly to daily or annual. And so this is also an improvement in being able to monitor the environmental conditions that might be occurring across all of these different regions. There's also been great advances in the availability and the quality of satellite imagery to look at landscape data, and also these really improved algorithms to be able to classify different landscape habitats. So previously, it might be possible to classify the landscape as agriculture or pasture. Now it's possible to really get to a fine scale and be able to differentiate things like fine scale or low intensity livestock areas versus high intensity livestock areas and other areas that might be relevant to this problem. And then with that, there's a lot more biological data resources available. So New World Screwworm is, livestock is the priority animal that everyone is thinking about, but there are other mammals and animals that it infects, and this can also impact its distribution and dispersal across different areas. So some of these newer resources, including even community science data, may be helpful in that area. And then Phil touched on this for just a minute, that there's quite a bit of advances in genetic data. So next generation sequencing, it's faster and more informative, or recently it's faster to acquire and more informative. It takes resources to be able to use next generation sequencing, but it has the potential, if there is genetic diversity across an area, to be able to map some of these populations of outbreaks across different regions. So along with all of this really vast, quantities of new data resources and access to different geographic areas, there's also been developments in in models that could be useful for New World Screwworm. And so when I'm thinking about the New World Screwworm problem, thinking about what's the overall distribution of the screwworm, the population dynamics, so when and where it might be in an area, and when would be the most optimal time to implement a control method, but also the dispersal. So where is it located now, and which direction might it go, or what is the easiest path for it to disperse so that it might be possible to set up interventions. So one approach is to use species distribution models. And so this is actually a species distribution model that was created by colleagues in Mexico, looking at using historical New World Screwworm outbreak locations and different combinations of temperature and precipitation. And then they predicted across this area where the environment might also be suitable for this species, given where it's been before. Then they kind of focused in on Central America and the southern US and overlaid their model with the density of different livestock across this area. And in this way, they were able to highlight different areas that could be sort of narrowed down toward high priority livestock regions that are also suitable environmentally for the fly. So this model could potentially be useful when trying to identify, general areas for monitoring or control, and that can help to narrow where resources are applied. So those models are really useful for overall distribution, but there's also this highly dynamic population component to the New World Screwworm fly. And because this fly has been studied so extensively in both the field and the laboratory, it means that there's a lot of parameters that are known about its development rates, its reproductive rates. And so this information can be used in combination with environmental temperature data to really try to take a look at when is the most optimal time to implement different control methods or eradication methods such as SIT. So this is an example of a paper and a model from Gutierrez et al. And they were taking a look across this environmental or temperature gradient at they retrospectively predicted these population dynamics of the fly, and then they compared it to the timing and dynamics of the SIT implementation to try to see whether or not there was a greater, when would it be most optimal? Is it optimal to use SIT in certain environments when it's really cold? Should it only be used in other environments when it can suppress the population more effectively? And you can see the reference here if you want to read that more in depth. That's not the greatest description of that model. However, there's also this dispersal component. And so kind of returning to Valdez Espinosa, they also included in their model dispersal. And that's, the host-mediated dispersal is a major component of this system. There can be short and long distance dispersal. This can occur because of densities of mammals or livestock across an area, as well as movement of animals and even wind events. And so here, this was a fairly straightforward dispersal model where they took different outbreak locations and then again used this livestock density surface to model these potential steps across this region. There are additional models that can be used for dispersal, including these connectivity models that use something called a friction surface. And so in the previous model, they were using livestock density as kind of this suitable area for possible dispersal. This model by Zaldivar Gomez, they were looking at something called a least cost path. And so they included some landscape and environmental variables such as altitude, distance to roads, and some other factors, including livestock density, to identify what would be the likely path of dispersal for the fly across this area. And so on the top is this least cost path algorithm. And then on the bottom is looking at potential dispersal into Mexico. And even though this manuscript came out in April of this year, this has already occurred. So there's also other dispersal models with different capabilities. So this is actually a connectivity or potential dispersal model that I created for Florida, looking at a hypothetical situation of Aedes aegypti mosquitoes dispersing from the Florida Keys, where there was genetically modified mosquito release. And here I used some population density data and these sampling locations to see what might be the most probable path. And so with this algorithm, it looks at all possible different pathways, and you can include different landscape or climate variables. And what's useful about this is that there's it identifies areas where there can be what's called these pinch points. And so it might be an area that's very narrow where there's probable dispersal, but it might not be very ideal for whichever species or organism you're interested in dispersing in areas nearby. And so this can help to identify where there could be intervention, or where you might want to also include additional monitoring or resources for surveillance. And then there's been updates to these where you can also have temporal time steps, and they can include things like mortality rates or other demographic parameters that are associated with the fly. So again, it's kind of coming back to this dispersal challenge is, that there is, it's not just livestock mediated dispersal. There's any mammal can be infested with New World screwworm. And so this kind of broad host range presents an additional challenge to trying to understand what is the potential dispersal of the fly in these different areas. And so I just wanted to touch back that there is kind of this growing set of biodiversity data that is available. And so that may be something that could be useful to be incorporating in the future, trying to look at this problem. And then the last thing that I'll talk about is landscape genetics. And so kind of going back to, again, what Phil was talking about with this next generation sequencing. Here, it's possible to go ahead and look at things, genetic differentiation or distance across different areas, and then use one of these path algorithms or models in order to map areas of likely dispersal. And so they were looking at genetic differentiation, different locations, and then they were able to use a species distribution model, and based on the environmental suitability, the map on the right shows the probable dispersal routes or connectivity between these populations, with deeper red areas indicating greater connectivity. So all of these data and models are really, great, but they don't really mean anything if they don't come back together in some sort of decision support tool. And so really the way that I see all of these tools and models is through this integrated pest management framework. And the purpose of these is to improve communication. So things like the dashboard could be quite useful for improving transboundary communication. Also, again, where do you optimize resources? Can these model outputs be useful for targeting different geographic areas or time periods? Can they help to identify areas where SIT might be more effective, targeting educational or informational resources, and also evaluating control. And of course, the overall goal of this is to reduce risk. There's not one model that's going to fix everything, or one set of geographic, tools or data. There's quite a bit of variation in resources, access, different components that will be require cooperation and organization across different regions to be able to combat this problem. So this is all that I have for today. With that, I can quit sharing my screen, hopefully, and turn it back over to Andrew. Here we go. Thanks, everyone.
Thank you so much, Dr. Campbell and Dr. Kaufman, for those presentations. I believe if there's an opportunity for those that are watching the live stream on YouTube to type questions, we do have a little bit of time for some discussion or addressing any questions that there may be, and I believe that Josh can route those to us if any come in.
We don't have any questions at the moment, right?
Josh, should we give it a little more time, or would you like to continue with the agenda?
I think we could probably move on, and if questions do come in, we can bring them in a bit later.
Cool.
If you'll be around, Phil and Lindsay. Great, thank you. Okay, let's move on to panel 2. So panel 2 is about sterile insect technique and new biotechnologies. In this panel, we've got Dr. Daniel Hahn, who's a professor in the Department of Entomology and Nematology at the University of Florida. He's been working with sterile insect technique in agricultural insect pests for more than 15 years. And we also have Dr. Alejo Menchaca, who's the director of the Animal Health Research Platform at the National Institute of Agricultural Research in Uruguay. He's directing a research program exploring the use of CRISPR-based technologies to control the screwworm. And without spoiling any more, I will hand over to Dan Hahn.
Thank you. Thanks for having me today. I appreciate the opportunity to speak. I'm going to share my screen and we'll get started. Can everybody see my single slide screen?
Yes, thank you.
Excellent. Thank you for the feedback. So as Josh said, I've studied the sterile insect technique for more than 15 years, and I've studied it primarily in fruit fly pests, moth pests, and then in the last 10 years or so, we've done much work on mosquitoes. I haven't actually worked on the new World Screwworm program, partly because it was hard to access here in the United States. And so I see this current outbreak as an opportunity to take some of the best things we've learned from emergent problems with mosquitoes, Mediterranean fruit flies, and moths around the world and integrate them into our understanding of how to make a more competitive, better quality sterile male so that we can have the best efficacy in the field and the greatest cost effectiveness. And so I want to talk a little bit about what we call a phase conditional approach to sterile insect technique. And it's how to really set up a program in a new place. And when I think about the screwworm in Latin America, whether it's countries like Uruguay or Argentina or Paraguay, all over essentially Latin America, this is a pest with a long history. And sterile insect technique has been applied there in small scales in the past. And I think we're really poised to the place where we could use sterile insect technique and other allied technologies to really make a big leap in control in the next decade. So when we think about sterile insect technique, there are 4 major ways in which it's used. The most common way worldwide is sterile insect technique is used to suppress pests. And in agricultural settings, for example, fruit production for fruit flies, where this is used the most, it's really used to keep pest populations low, to reduce insecticide use, and have low pest prevalence to enable movement of fruits and vegetables outside of areas where the pest occurs. To me, this sounds a whole lot like the problem with cattle. We want to make sure that we're suppressing the pest so that we're not infesting cattle, but also allowing export and movement of cattle. The technique is used for containment to avoid the spread of pests from one area to another, prevention to avoid establishment of pests, and of course, eradication is really often a big goal for any program. We would love to rid all of the Americas from the New World screwworm. And I think, though, that eradication as a goal is often a really lofty goal. In the past, the United States, in partnering with Mexico and other countries, was able to eradicate the New World screwworm. It started, of course, here in Florida. We had great success getting rid of it from Florida and the Southeast, and it marched across. Most of us here will know this story. And then, of course, beyond the eradication from North America all the way down into Central America, for 20 years or more, a sterile insect technique was used to prevent the pest from migrating back up. But I don't think this is the only model. Here in California, Florida, elsewhere in the world, we also use sterile insect technique to prevent the pest from emerging in a new area. For us, we often use this for invasives that we want to keep out. But really, what I think is going to be most effective in the short term is not thinking about eradication. That can be a long-term goal. And one of the things we were asked for today is short, medium, and long-term goals. I think a medium-term goal for regions in Latin America is to really think about local suppression before eradication. And local suppression is used frequently in sterile insect technique programs. I've been fortunate to be involved with a number of local suppression programs here in Florida for mosquitoes, also with mosquito suppression programs worldwide. And I think this is really the model for a medium-term success, with eradication being the really long-term model. So eradication is not always the thing we need to think about first. Now, when we think about sterile insect technique, there are a number of different flavors of sterile insect technique, if you will. When folks in the field think about it, we often think about the traditional radiation-based sterile insect technique, where we use either gamma rays or X-rays to sterilize flies built up in a factory, and we send them out into the field. In the current situation with New World screwworm, a major downside of this technique is we currently cannot separate males from females easily. And so you end up paying the costs to grow roughly 50% females, and this leads to lower program efficacy. There are other techniques that have been used in other insects that I think are highly applicable to the screwworm problem. There's incompatible insect technique. The most popular form of this is induced by infection with a bacterium called Wolbachia, but there are other bacteria and other ways to induce incompatibility. A major downside of this technique is that if females are released, they could cause replacement of the population, and then you lose efficacy. And I believe the next speaker is going to talk about genetically modified options for sterile insect technique. And I do think these are part of the future. In fact, our lab builds genetically modified sterile males ourselves, and we do this in fruit flies. But in the short term, getting genetically modified technologies in the field can have some serious challenges, particularly with public perception. And with any genetically modified technique, one needs to really think about the risks of establishment of these genes, these transgenes, into the environment. So I'm going to focus today in thinking about our project here as radiation-based sterile insect technique, because it's what we have today, and it's really actually very successful if you can get it to use it in the right conditions. There is an existing framework. So many of you may know that the United Nations Food and Agriculture Organization partners with the International Atomic Energy Agency, and they have a whole section called the pest control subsection that applies peaceful uses of radiation to agriculture. Part of this group's purpose is to support the sterile insect technique in all United Nations member countries. As part of this, they have produced some really nice documentation, particularly for mosquitoes in the last decade, that lays out how to successfully set up a sterile insect technique program in a novel area. And it is this so-called phased conditional approach. It has to start with pre-intervention, really surveying for potential sites. And these are where things like geospatial data, like Lindsay talked about in the previous speaker, come in handy. How do you choose an appropriate site? Baseline data collection. How do you know what the pest populations are in that site? And that can then lead to things like preliminary field trials. If those preliminary field trials go well, you can then move all the way up to the top of the pyramid where you have an operational program. And I think it's really important. Sometimes when folks are new to sterile insect technique, they believe they can jump very successfully straight to an operational program. But like any other form of integrative pest management, you need a basis of information to build on. Now, fortunately, over the last 20 years or more, there has been much of this base built up already. So there have been great collaborations between the countries of Brazil, Paraguay, and Uruguay. The International Atomic Energy Agency working with USDA and COPEG and the International Development Bank to start thinking about this problem. So we already have some of this basis laid out. For example, there has been baseline data collection done in a number of places in Uruguay, Argentina, Brazil. There's really pretty good data from this project done in 2007 to 2009. And I think some of what would need to be done is to revisit those data and to think about how to best use those data. There are great sites that were already picked during this time period, and we could think more about potentially, are those still the most appropriate sites? I think one of the things to always think about when you're considering sterile insect technique programs, and it is a cornerstone of this phase conditional approach, is where is the financial support for sterile insect technique to come from? And I think there's a lot of different models. If you look at sterile insect technique programs across the world, some are funded by one central government, many are funded by government entities coming together and working together, like the current COPEG facility in Panama. Some are public-private partnerships where growers themselves contribute funds to additional supplementary funding from governmental agencies, and you get a partnership that can pay for the sterile insect technique. And some, including fruit fly and sterile false coddling moth programs in South Africa, are paid for almost entirely by producers. It's a commercial model there. So you can think about many different models, and which model one chooses really depends upon the region and what kind of support is available. But I think it's really good to think about all these different models, because you can see that across the world, sterile insect technique has been successful with a variety of funding models. Now, when we think about medium-term priorities, again, some of these have already been completed. There was a fairly successful pilot field trial conducted for essentially 4 months between USDA, COPEG, and the countries of Brazil and Uruguay. And this was done between 2007 and 2009, with the majority of the releases occurring during the summer of 2009. And what we saw is over 4 months in these data, there was successful dispersion of flies in the field, and in some weeks up to 40% suppression of the local pest flies. And this really, I think, showed that there is feasibility for this local suppression model. Now, I think the program in retrospect would have been more successful in 2009 had they started at a different time of year. They started at the summer, which is, of course, the time of peak pest populations, and sterile insect technique works best when pest populations are low. We spend a lot of time thinking about how do we get males out into the field early enough before pest populations build to get effective control. And also, of course, you have to have large numbers of flies. But I think with more planning and more infrastructure for fly production, this could be even more successful for suppression of local populations in Latin America. Now, when we think about the sterile insect technique, of course, there's a whole series of steps. There is an entire pipeline of production from strain development to successfully mass rearing those strains. In an ideal situation, you can somehow separate out the females from males, and this is currently a bottleneck in screwworm. You have to sterilize them, you have to perform quality control, you have to package them, transport them to the field, release, and follow up with field monitoring. And sterile insect technique programs have to be strong at each of these levels of the workflow to be successful. And what we focus on in our research group here is consulting with sterile insect technique programs to maximize their ability to do each of these steps successfully and to maximize what we call both process quality control, which is how effectively, cost effectively do you make the sterile insect, and then also product quality control. Is your sterile insect of the highest quality? Can it mate compatibly with the wild females in an area where you're trying to get suppression? How effective is this? And by maximizing both process and product quality control, you can make sterile insect technique programs both effective and affordable. And this is what we focus on in our lab with mosquitoes, fruit flies, and moths. Now, these steps in both product and process quality control in the last 20 years in the US have been led by our researchers in the USDA ARS and USDA APHIS laboratories, particularly the group in Kerrville that Phil Kaufman mentioned. They have really led the charge on this in collaboration with their staff at COPEG in Panama. But there are other folks, and I think the place where universities can really contribute is thinking about which of these steps can we help these operational programs maximize their ability in. And so how can you have the best possible strain for your particular area? It's really important that the males you're releasing can mate very effectively with the wild females. And if you pick the wrong strain, you'll have less effective pest control in the field. Mass rearing, how can we affect the efficacy of mass rearing? People are working already on the sex separation problem, and I won't talk very much about it because the next speaker is focused on genetic technologies. But Max Scott at NC State has collaborated with folks from USDA and also the group in Uruguay who will be speaking next to think about various transgenic methods to produce males only, as well as genetically modified methods for sterilization. My colleague Aaron Terone at Texas A&M has worked a lot recently on new process control for sterilization. And I think where university sectors like us can really play a role is coming into programs and helping with these workflows to make them more effective and more cost efficient. And so when I really think about what's the best strategy here, and we were asked for short, medium, and long-term goals, when we look at the eradication program in North America and you look at the historical development of this, it was really local suppression and eradication in Florida and the Southeast that allowed the program to gain enough momentum to really push for eradication across all of North America and eventually Mexico all the way down to Panama. The Texas cattlemen saw the Florida cattlemen saving money once Sterile Insect Technique and other integrative pest management strategies were used together to get rid of screwworm in Florida. And I think this would be one of the best models to move forward, is to choose an area, a smaller area, perhaps that's more isolated, and you could effectively get suppression once producers in that area start to see a financial benefit from control. Other producers will want this as well, and that's how you gain momentum. So I think a real medium-term priority would be to have a successful suppression program somewhere in Latin America. And we know that while the fly is currently widespread, as Lindsay talked about in her presentation just a few minutes ago, we also know that there are corridors for movement of pests and there are barriers. For example, there was a very nice study done on the border here between Uruguay and Argentina, where they released sterile marked flies on either side of this river, marked with different colors of marking, and they found no passing of flies across the Uruguay River. And so you can use natural barriers and areas with lower densities to choose a proper area and get some success. Once you can really get some success and the producers in that region see the economic benefits, I think it's critical to trying to spread any of these control strategies more widely. And so when I think about long-term suppression of New World Screwworm in South America and other places in Latin America and the Caribbean, we all know, as has been mentioned before here, that sterile insect technique is one tool in the toolbox. We need strong integrative pest management. We need livestock management. We need management of flies in many different ways, but that if you have these integrated management schemes, sterile insect technique can be a really strong tool for suppression and perhaps even eradication. But the key, I think, really is to continue to innovate. And new innovations are, I think, poised to help the screwworm programs have greater efficacy and greater cost efficiency. And I think that's what we really need to focus on from the university side, that we can translate out to active programs, start small and build success in a contained area. And then, of course, one needs to work out dedicated long-term budgeting from commercial and nonprofit and government sources, because to have a really successful sterile insect technique program, you've got to have dedicated space and staff. Now, there are, of course, bottlenecks, but technology is really allowing us rapidly in other sterile insect technique programs, particularly mosquito programs, to jump through a number of those bottlenecks. And I believe this is equally as possible to make more progress in screwworm sterile insect technique over the next 5 to 10 years than's been made in the last 30 years. And our key is to really be flexible and responsive and consistent despite setbacks, because if anybody who's worked out there on integrative pest management knows, there are always setbacks like the one we're experiencing with the current outbreak. So I want to thank all of the folks who have supported our work on sterile insect technique in the recent past, including funding from many different sources, our colleagues at the United Nations International Atomic Energy Agency, FAO, Pest Control Group, and our friends, particularly in mosquito control, that we've worked on in the last 5 years. So thank you.
Thank you very much, Dan, for that very informative overview. It's great. I will pass the floor over to Alejo Menchaca.
Okay, thank you. I'm trying to share the presentation. Can you see it?
We can't see it in full screen yet.
Okay, thank you very much for the invitation. I'm looking at the YouTube channel. I see a lot of people from Latin America countries, so I think that will be better to speak in Spanish. FONTAGRO has the interpreter to translate the presentation. So, entonces, si me permiten, voy a hablar en español. Para los habla hispanos va a ser más fácil, seguramente, poder seguirme. Entonces, esta pequeña charla tiene que ver con el potencial aporte de las tecnologías basadas en CRISPR para el control del gusano barrenador. Es un trabajo que venimos haciendo desde Uruguay, desde el Instituto Nacional de Investigación Agropecuaria, INIA, en conjunto con el Instituto Pasteur de Montevideo. Y de alguna manera quiero compartir algunas lecciones que hemos venido aprendiendo durante estos años desde el sur, desde nuestro país y en nuestra región, compartida con Argentina, Brasil, Paraguay. Nosotros estamos en una región que produce mucho alimento. La producción bovina para nosotros es muy importante desde el punto de vista económico, social y también desde el punto de vista de la sostenibilidad ambiental, porque es un sistema bastante equilibrado. El 35% de la población bovina del mundo está en las Américas y producimos aproximadamente el 50% de la carne que se exporta. Entonces, producimos mucho alimento, tenemos una ganadería con mucha densidad, sobre todo en el sur, como se aprecia acá. Cuanto más oscuro el color, más rojo, mayor densidad de ganadería. Y entonces, este escenario, de alguna forma, se combina con la presencia de la mosca de la bichera o el gusano barrenador, como le llamamos en estos países. Así que tenemos, de alguna manera, la necesidad de convivir produciendo alimentos, pero a su vez conviviendo entonces con una plaga para la ganadería y para la sostenibilidad, sobre todo productiva, con este parásito, con esta enfermedad. Uruguay entonces es un país absolutamente ganadero. Por cada habitante tenemos unas 6 hectáreas en promedio de campo, y en esas 6 hectáreas tenemos 4 vacas, 2 ovejas, tenemos caballos, es decir, tenemos una población de ganadería muy intensa, y entonces trabajamos desde la investigación y desde la producción enfocados en el abordaje una salud. Tenemos que considerar la producción animal, pero también teniendo en cuenta el resto de la salud pública y del ambiente. Así que cualquier cosa que hagamos y que estamos haciendo y estamos trabajando, estamos contemplando los diferentes componentes de ese concepto de una salud. Cuando hablamos de gusano barrenador del ganado, en todo Sudamérica y yo diría hoy en toda América, genera problemas realmente graves. Tenemos, por un lado, un problema vinculado a la sostenibilidad ambiental. Se utilizan pesticidas actualmente para controlar este problema en todos nuestros países. Todos los países de México al sur utilizan pesticidas o insecticidas para controlar este problema. Por otro lado, afecta desde el punto de vista económico, hay un gran perjuicio con pérdidas cercanas a los 3.5 billones de dólares año tras año. Genera un problema grave y muy serio de bienestar animal, tanto en los animales domésticos, pero peor aún en la fauna silvestre. Los animales domésticos podemos tratarlos con insecticidas, pero los animales silvestres no pueden ser tratados y por lo tanto terminan sufriendo y muchas veces mueren en una agonía prolongada. Además de esto, este problema para nosotros, para todos estos países, también tiene algún vínculo con la salud pública, porque en algunos casos también puede afectar humanos, como ha sido la situación recientemente en algunos países de Centroamérica y Norteamérica. Entonces es un problema realmente grave en muchos sentidos. Hasta ahora, digamos que hasta hace poco tiempo, el problema estuvo de alguna forma limitado al sur de América y en estos países ha sido y sigue siendo probablemente el principal desafío que tenemos del punto de vista sanitario vinculado a los animales y afectando también en otros aspectos. La erradicación en estos países ha sido propuesta de diferente forma, o el control, además del uso de insecticidas. Ustedes, como bien decía Daniel en la charla previa, todos ustedes conocen la técnica del insecto estéril, que primero se aplicó con mucho éxito en la década de los 60 y en los 2000 llegó a lograrse en Panamá, generando la barrera en Darién. Llevó unos 40 años este proceso, erradicar en toda esta región. Entonces, cuando uno mira hacia el futuro y piensa que hoy de vuelta la situación está volviendo al pasado, lograr nuevamente esto, si antes llevó 40 años, yo no sé cuánto podrá llevar en el futuro, pero seguramente va a llevar muchos años volver a la situación que teníamos hace un par de años en la barrera de el Darién en Panamá. Por lo tanto, cuando nosotros consideramos, por lo menos en Sudamérica y en la región en la cual nosotros vivimos y convivimos con este gran problema, tenemos que saber que hemos convivido siempre con él. Este insecto está acá antes que llegaran las vacas y hemos considerado también la erradicación por la técnica del insecto estéril. Uruguay pierde 50 millones de dólares aproximadamente cada año por este problema y por lo tanto está en el foco de atención del gobierno, de las autoridades oficiales, de los investigadores, de los veterinarios y de los productores ganaderos. Es un tema realmente importante. Entonces, cuando nos planteamos el uso de la técnica del insecto estéril en nuestro país, realmente ha sido un gran desafío poder aplicar esta tecnología. Tenemos un territorio pequeño, pero con una frontera muy amplia. Nosotros tenemos frontera con Argentina y con Brasil. Es una situación inversa a la de Panamá con Colombia. Mientras en Panamá y Colombia hay una frontera de 100 y pico de kilómetros, en Uruguay tenemos más de 1.500 kilómetros de frontera, siendo un país muy pequeño. Entonces, además tenemos la presencia endémica de este problema en los países vecinos. Requiere esta tecnología una liberación permanente y eso implica muchos esfuerzos y recursos económicos. Y hasta ahora la facility, digamos, la planta para producir esta mosca estaba únicamente en Panamá. Entonces, también esto, si uno mira las fortalezas y las amenazas y las debilidades de avanzar por este camino, esto era una debilidad y probablemente una amenaza. Hace 2 o 3 años o 4 años cuando se estuvo estudiando la posibilidad de aplicar esto en esta región del sur. Y demanda, por lo tanto, políticas de Estado a largo plazo, lo cual también es difícil asegurar en los países de Latinoamérica. La situación ahora es peor que hace 3 o 4 años porque, bueno, todos sabemos la situación en la que nos encontramos. Entonces, la mosca, el insecto, avanzando hacia el norte y probablemente, por más que tratemos de comprender y entender cómo es este proceso, lo cierto es que las herramientas para controlarlo y volver a la situación anterior es un gran desafío. Entonces, nosotros creemos que tenemos que buscar nuevas alternativas, seguir utilizando las herramientas que hasta hoy tenemos, pero tenemos que buscar herramientas que sean más precisas, más ajustadas al problema que estamos teniendo. Ese es el concepto en el cual estamos en Uruguay desde hace ya unos años trabajando en esto, básicamente con el uso de tecnologías basadas en CRISPR. Hemos aplicado esta tecnología y la estamos estudiando desde hace unos 10 años, 10, 12 años, 10, 11 años, trabajando en CRISPR, y hace unos 4 o 5 años que estamos trabajando en el gusano barrenador. Ha sido un camino difícil, le voy a contar muy brevemente lo que hemos venido haciendo, pero bueno, para los que no están tan involucrados en el tema de CRISPR, de esta nueva herramienta, es una herramienta que la hemos venido aplicando en ganadería para diferentes usos, para mejorar la productividad de los bovinos, por ejemplo, para generar animales que sean resistentes a ciertas enfermedades, sobre todo virales, pero también para el control de enfermedades transmitidas por vectores o incluso para el manejo de ciertas plagas para la ganadería o la salud pública, y también en el uso contra la lucha de especies invasivas en algunas condiciones, y probablemente surjan otras aplicaciones en el futuro. Pero si nos queremos enfocar en el control de plagas mediante esta tecnología, ahí es donde creemos que podría ser una contribución y complementar probablemente otras herramientas que hemos venido usando. No estamos pensando en sustituir absolutamente todo el resto, sino complementar y sumar más herramientas a la lucha contra esta dificultad. ¿En qué consiste el uso de gene drive mediado por CRISPR para lograr este objetivo? Básicamente, mediante el cruzamiento natural que ocurre en una especie silvestre como el gusano barrenador, una característica fenotípica determinada por la genética se perdería rápidamente en una población. Si nosotros queremos diseminar una cierta característica en esta población, que por ejemplo permita controlar la población, sería absolutamente ineficiente, ineficaz, porque se perdería rápidamente. Sin embargo, mediante Gene Drive, en la medida que nosotros podemos incorporar a nivel genómico y en una secuencia determinada, en un alelo determinado, cierta característica o una edición que nos permita una característica fenotípica que logre que por ejemplo, que las hembras no sobrevivan y solo los machos sobrevivan, esa característica se va a ir transmitiendo mediante esta tecnología de manera dominante, por así decirlo de alguna forma, de generación en generación, y las hembras seguirían siendo suprimidas y entonces los machos seguirían transmitiendo esta característica una y otra vez, generación tras generación, de tal manera que en pocas generaciones esta característica estaría absolutamente expresada, diseminada en esta población silvestre y podríamos controlar de una forma absolutamente distinta esta población silvestre, controlando el problema y la cantidad de moscas que nos están afectando. Esto es lo que estamos haciendo en Uruguay. Para eso, nada más que mostrarles el trabajo que nos ha venido dando generar esta nueva herramienta. Realmente no es algo simple que se genera de un día para el otro. Se requiere muchas cosas, se requiere cooperación, se requiere inversión, recursos, en fin. Nosotros estamos en Uruguay, entonces, acá en esta imagen se ve la facility en la que trabajamos, donde yo estoy en este momento dando esta charla. Tenemos entonces un bioterio entomológico que hemos construido. Generamos una colonia en cautiverio de Cochleomia omnivораx, de esta mosca. Tenemos estas condiciones para producir las moscas en cautiverio, que también fue un buen aprendizaje. Generamos laboratorios, equipamientos, entrenamos investigadores que trabajan en nuestro equipo. Hemos hecho muchos, digamos, acuerdos y estamos trabajando en conjunto con Max Scott como consultor o como advisor nuestro también. Y todo el equipo entonces inserto en Uruguay trabajando de alguna forma para resolver nuestros problemas. Hoy tenemos la capacidad de producir este insecto para trabajar en investigación y generar entonces el modelo que estamos procurando, que estamos buscando. Básicamente, entonces hoy lo que hacemos cada día, todos los días de la semana, los investigadores con los que trabajamos juntos acá, es producir embriones de esta mosca, inyectar estos embriones con técnicas basadas en CRISPR y generar entonces moscas con estas características. Entonces, en lugar de que el problema, como habitualmente lo veíamos en el campo, hoy podemos reproducir esto y hacerlo directamente en nuestro laboratorio sin ninguna dificultad. Un trabajo que nos ha llevado varios años. Entonces, la tecnología que estamos desarrollando es un gene drive de supresión. Para eso, bueno, esta herramienta tiene varios componentes, hay que irlos generando y diseñándolos y armándolos y viendo cómo funcionan en forma secuencial. En eso es en lo que estamos hoy trabajando. Y básicamente consiste en la técnica en tomar los huevos embrionados, inyectar la construcción con CRISPR y después entonces esperar y ver que efectivamente funciona primero en las larvas y después en las moscas adultas. Actualmente hemos avanzado con el equipo en generar hembras infértiles que no se pueden reproducir. Esta foto muestra entonces un macho, una hembra y la diferencia que hay fenotípica entre la hembra y el macho. Aquí se ve, bueno, y un ovario de una hembra fértil y un macho fértil como produce espermatozoides. Sin embargo, cuando nosotros editamos ciertos genes de interés vinculados a la reproducción de estos animales, de las hembras sobre todo, se puede generar un fenotipo infértil y por lo tanto las hembras no se pueden reproducir. De esa forma, todas las hembras que nacen dejan de multiplicarse y los machos, en cambio, siguen transmitiendo esa característica cuando se cruzan con una hembra silvestre, con una mosca silvestre. Así es como esto rápidamente podría ir resolviendo el problema a mediano plazo. Entonces, para terminar, simplemente contarles que este es un proceso que no se resuelve de un momento para el otro. Yo creo que si, dada la situación actual en el norte de América, si se fuera por este camino, seguramente va a llevar varios años de trabajo. Tenemos con la cooperación técnica, como bien lo mencionaba FONTAGRO, tenemos la posibilidad de ofrecer desde Uruguay por lo menos 5 años de camino ya andado y probablemente podamos ahorrarle a quien quiera atravesar este camino, ahorrarle un buen tiempo si pudiéramos con gusto contribuir desde nuestro lado, desde el sur. La primera fase en la que hemos estado trabajando entonces ha sido en todo esto que dice acá, para generar entonces moscas con esta característica. Vendrá después una fase de validación para finalmente, después de algún tiempo también, importante, poder tener un producto, una herramienta absolutamente disponible para que se evalúe la posibilidad de utilizarla en un programa nacional. Ese es el camino, estamos en la primera fase, por lo tanto las expectativas hay que manejarlas con prudencia y al mismo tiempo hay que ir trabajando en estos puntos que aparecen acá abajo, obviamente con la cooperación científica entre los diferentes países, con los recursos necesarios para poder invertir lo suficiente y avanzar lo más rápido que sea posible. Para eso tenemos, como decíamos, hemos articulado a varios intereses para avanzar en conjunto, desde acá, desde Uruguay, con financiación prácticamente local de nuestro país, el apoyo del Banco del BID, también del Banco Interamericano de Desarrollo, y a su vez realizando estudios de impacto ambiental, monitoreando la población, todos aspectos que hay que ir considerando también al mismo tiempo. Y a su vez, nada menor, desarrollar y diseñar un sistema regulatorio apropiado para poder usar estas herramientas con seguridad. Para eso también es importante involucrar a todos los actores del gobierno que de alguna forma velan para poder usar herramientas cada vez mejores. Así que todo esto lo hemos articulado y está funcionando de acuerdo a lo planificado y ese es un poco lo que quería compartir con ustedes y mostrar de alguna manera lo que hemos aprendido durante estos años y lo que estamos aprendiendo trabajando para mitigar esta problemática. Muchas gracias por la atención.
Muchas gracias, Alejo. Excelente la presentación. So, Dan and Alejo, I think we have some time for questions before a short break. So I think, well, Dan first, considering we've heard these options of sterile insect technique and novel genetic control techniques, how do the timelines to deployment for deployment differ between these different techniques?
Yeah, thank you for asking that, Josh. As Alejo said really nicely, gene drive techniques have been developed in a number of organisms, and their group is doing a great job of developing it in New World Screwworm. But at the moment, it's still at the R&D phase, and it's going to take some additional time and investment to get out of that initial R&D phase to a deployable product. And not only will it take some time, but there will be substantial potential regulatory concerns that even once the product's ready to go out, may slow its deployment. I think genetic techniques like gene drives are certainly part of the future of control. And I strongly encourage them with our collaborator, Al Handler at the USDA here in Gainesville, Florida. We are also working on gene drives in fruit flies, but you can't have it working today. And so right now, what we have currently available is sterile insect technique of the traditional method. Now, Alejo also brought up a really good point, and that is we don't even really have sterile insect technique for Latin America today because there isn't enough current capacity at the Copeg plant. And as Alejo properly said, it's probably going to be a number of years before we can solve the problems with New World Screwworm invasion into Mexico and outside of the Darien Gap. But I think once we can solve that program, there will be capacity. And I don't see these as opposing technologies. I see them as complementing technologies. And that is because gene drive, even though you will have supra-Mendelian inheritance in the population, you have to produce enough of the animals initially, and you have to get them out into the field. And into the field, ultimately, in many locations. We learned a lot of lessons about this from the Eliminate Dengue program in mosquitoes that has been implemented in many places in Asia and a few places in Latin America. We know that establishment of a supra-Mendelian inherited trait takes a lot of input of animals. And so I think these two are complementary. Certainly what Alejo's talking about is the future, and I hope the very near future. But I think both of them work together and can be used complementarily. Yeah, so you have to have process quality control and product quality control of your gene drive flies before you send them out into the field. And that's some of what we work on here as well. So thank you for that question, Josh.
Thank you, Dan. Alejo, ¿algún comentario?
Yes, thank you, Dan. I agree absolutely with Dan. I think that, of course, we are working here, but we are thinking to work together with the other techniques. And Uruguay is thinking to use the technique of sterile insect, and probably in the future, in the near future, we can apply these conventional tools, but also we are developing at the same time the other tool for the future. So I think that's the complementation of both could be the best choice to work together. Yeah.
So following up on the development of CRISPR techniques, Alejo, bueno, no sé si quieres la pregunta en español o inglés, pero ¿conoces si hay avances en el tema de regulaciones que permiten o autorice el uso de estas tecnologías? ¿Y cuáles crees que son las barreras institucionales o societales principales para superar?
Ok, bueno, respondo en español. Creo que el público principal es en español. Sorry for the other people that is speaking in English, but I think that the translator is working well. ¿Funciona bien? Yo usé la traducción al español, al inglés. Bien. Entonces, yo creo que por lo menos en nuestros países, en el sur, los temas regulatorios van acompañando el peso del problema, el peso de la problemática. Yo entiendo que es difícil acompañar el avance científico desde lo regulatorio cuando los problemas no son muy graves, los problemas que solucionan esas herramientas. En la medida que el problema es más grande, el sistema regulatorio trata de adaptarse, digamos, de avanzar rápido también. Entonces, una cosa es un sistema regulatorio para un problema que no existía en Estados Unidos, como hasta ahora. En el otro extremo estamos Sudamérica, que teníamos este problema grave. Y otra cosa es entonces cuando ahora el problema ya está llegando al norte. Entonces, pienso que estas cosas van a ir cambiando con el tiempo y ese tiempo se va a ir acortando en la medida que el problema va siendo cada vez más grande. Por supuesto que el sistema regulatorio tiene la función de velar por que las herramientas sean eficaces, inocuas, contemplando todo el contexto de una salud, no solo lo animal, sino también lo ambiental. Pero bueno, por lo menos en estos países del sur estamos en ese camino, intentando tener una regulación acorde al problema y considerando obviamente los riesgos también.
Muchas gracias, Alejo. And Dan, well, same question to you for the US. Have you seen any advances in regulations and how that may be relevant as well to Latin America?
Absolutely. And I agree with what Alejo said. When the problem gets bigger, the pressure gets bigger, and regulation is more likely to move faster. We have seen this in disease vectoring mosquitoes, where these emerging technologies, including genetically modified options, are gaining much more rapid regulatory acceptance and public acceptance because of the pressure from, say, dengue virus infection or the Zika virus infection that swept across much of the world a few years ago. This really showed us that you can move things fairly quickly regulatorily. And so I don't think we'll be in a case where we work together to make a tool that will never be deployed because of regulation. I believe that in the United States, we have seen regulatory landscapes shift to be more favorable towards genetically modified options. CRISPR-based gene drive is at the very cutting edge of this. But I believe if we lay the groundwork now, this tool can be used much more rapidly than perhaps previous genetic technologies were deployed. And so I am actually very hopeful about this section of the future in the US and abroad, particularly in Latin America, where the pressure for this pest is so strong. I think regulatory hurdles will be smaller, and I think the landscape of regulatory hurdles in the Americas is very different than, say, it would be in Europe. Much more favorable.
Excellent. Well, thank you both for your excellent presentations and your
Thank you
thorough
very much.
answers. And thank you, Eugenia, for the question on regulations. So now I think we're going to get ready for a short break, about 5 minutes. And I would ask at the end of the meeting if everybody can stay on for a group photo. And I'm pleased to say that after the break, we will have a short presentation from Dr. Justin Talley from Oklahoma State University. Thank you for joining us, Justin. So I will turn off our cameras and we can resume in 5 minutes. Okay? Thank you very much.
Hey, George, can you hear me?
Yes, I can hear you.
Okay. I was trying to ask a question. I don't know if you heard me then.
Oh, I'm sorry. No, we didn't hear you. We can absolutely take that question after the break, if that's okay. My apologies, Lola.
Okay, that's fine. That's fine. Thank you. No worries. No worries.
I think just before your presentation, Justin, I had a question for the panelists. Okay.
Hey Josh, I can't hear you very well. Can you hear me?
Yeah, Josh, your audio is breaking up.
Okay.
How's that now?
Better.
I was just saying that before your presentation, Justin, I know Bola had a question for Alejo and Dan, if they're there. If not, we can leave it for the closing session.
I'm sorry, I just stepped away to get a drink of water and I just heard my name as I was coming back to my office. Do I have an action item?
I know Bola had a question that I didn't get to at the end of your panel. You're muted, Bola.
Can you hear me now?
Yes, I
Yes.
can hear you.
Okay. Sorry, trying to get rid of the echo. Okay, my question is about whether there are concerted efforts to bring experts together from the US and from Latin America to work on this threat. We know about the collaboration between Texas A&M and I think it's Embrapa and maybe Uruguay, and then Uruguay, North Carolina. But a problem of this nature seems to me to require bringing together experts in a much more concerted and strategic manner. We know that FONTAGRO is funding some groups, and this whole meeting is because of FONTAGRO. So thank you, Eugenia. But it just, as I listen to all the expertise, it just seems like if we had all these experts working together, we could make a lot more progress quickly. Thank you.
Thank you, Bola. I'm just want to make some comments. First, I really want to thank all the presenters. I'm taking note, and it's really interesting how really this webinar bring the idea of an integrated approach is needed. And it's not just what we can do with livestock in the field, it's also one health issue as well, as this can affect or impact on public health as well. So I think the sharing the views of and the approaches of every scientist, I think is very important, because we need to have an idea on how to tackle this challenge from different perspectives and different experiences. I love what Daniel says about all his experience and all the track records historically in the US, for example, and also Lindsay mentioned and map and show the mapping on how that was evolving. I'm not sure in Latin America how much we have done apart from Argentina and Uruguay, which are, and maybe Brazil, because they are the two main livestock producers. But I think that many other countries as well, I mean, every country has livestock. So these have to be something that we need to work in a collaborative way, Latin America with the scientists as well, and the offices in other countries like Mexico, Brazil, and the US. I don't want to take a lot of time, but the two aspects that I will like to highlight right now is the integrated approach, public and private, the research institutions with the national animal health offices as well, and the private sector. And when we are talking about the private sector, we are not talking about only the farmers associations. I'm talking about the food system related to the cattle and livestock and the beef industry itself, and also the transport industry. I mean, every time that we move animals, we need to have some kind of protocols that it's not clear to me right now is that exists, or we need to work on that as well. And I'm saying that because I was working in ranching for a long time. And the second point that I like to highlight by now is the importance of innovation and how different scientists can bring their expertise, knowledge, and the challenges they have on developing new innovation and new knowledge on this. The sterile insect techniques are very important. At the same time, the ICT techniques are very important because we need to be better at detecting earlier where these pests and disease kind of starting to become a real problem. And something that I would like to point right now and maybe take it later is the animal identification and traceability systems in livestock. This is something that I was working for a long time as well. If we have a clear idea how animals are moving, and in South America, that is very complicated because, as Alejo mentioned, we have 1,000 kilometers that we are neighbors. We work as a neighbors, which is different between Panama and Colombia. But at the same time, I mean, animals are moving, people is moving, and we need to see if there are some protocols that we need to develop every time that we move animals and in between borders or even in between sub-regions inside a country. So, voila, I think the work that we are doing is very important. I think it's one of the first time that we are putting together all the best experts in this topic to talk about how we can tackle this important challenge that we have as an industry. And let's see what the next panels will bring to us as well. And let's pass to the closing remarks at the end. So thank you so much, everyone. It's really important what we are doing. And thank you, Andrew and Josh, for coordinating with us this webinar. So I pass to you, Josh. Except that Andrew want to mention something.
I was going to pass to Andrew.
No worries. Thank you so much. Yes, I just wanted we're going to have a little addition to the schedule before panel 3. My colleague Justin Talley is the chair of entomology and plant pathology at Oklahoma State University, has joined us. And Justin has some experience doing some capacity building in this space. And he's going to give a little presentation about training related to New World Screwworm. Thanks, Justin.
Thank you. I'm going to try to share my screen and get through this fairly quickly. Can you see? Let's see. I got the wrong.
We can see that.
Yeah, it's all good.
You can see the presentation?
Yep.
Okay. Good. So just as Angie said, I'm the department head for entomology and plant pathology at Oklahoma State University. I'm also a veterinary entomologist by training. Phil Kaufman and I run in the same circles, if you want to call it that, in the work that we do. When we think about workforce development, it's really about forming partnerships. The partnerships that we really want to kind of highlight are, what are veterinarian entomologists. And I just took a quote out of something that was published here in the American Entomologist by a pretty well-known veterinary entomologist, Brad Mullins. And by just a rough estimate, we've lost, several capacity in veterinary entomologists that are at academic institutions. Now, we've seen a little bit of an upsurge here recently, and it may be due to the New World Screwworm in positions. But, the thing that I really want to highlight is, what does the veterinary entomologist bring to the whole dynamics of understanding New World Screwworm? Well, it's not only the biology and ecology of the pest, but also how that host-parasite relationship forms. But also, we also have the training where veterinary entomologists understand production systems. Veterinarians are broadly trained, and rightly so, and they do a really good job at attacking problems when they need to. But sometimes the details in what matter in knowing the difference between Cochleomya macellaria and Cochleomya hominivorax is important, and that's usually done by veterinary entomologists. And I would like to point out, veterinary entomologists developed the sterile insect technique that was used for the screwworm. And I just took this from the FONTAGRO, one of their reports, and how everything they do is related to UN sustainability goals. And why I bring this up is because we took some students down to the COPEG facility, undergraduates and graduate students, just to get broadly exposed to a facility that does phenomenal work. And I just wanted to highlight, when we think about workforce development, FONTAGRO seems to already have the infrastructure where we could plug in. And the thing I just want to highlight is, there's a lot of livestock projects going along into a lot of different countries, which have been highlighted already. What the goal is to provide an educational value, an experiential learning experience, and an interdisciplinary opportunity. Again, we want to build capacity. We want to build capacity in the form of people that can critically think about the problem as the New World Screwworm presents, both in Latin America and in the US. And then we want to kind of form a framework where we can really shape this model into a high impact model. Some of my experiences in capacity building are related to both veterinary entomology training as well as biosecurity training. Another hat I wear, I run an institute called Institute of Biosecurity and Microbial Forensics, and we are currently training several graduate students in global ag biosecurity. So the main thing is linking it to governmental stakeholders. It's like we've got to link this to national biosecurity, regional stability, and agricultural resilience. Again, we're not just putting students out there to be exposed, we're putting students to be boots on the ground so they can help their community. Again, the structure of this is they're going to be exposed to several different things, or what we would like to propose is that they're exposed to several different things, and that we're just eager to work with. And like Dr. Coffin said, Texas A&M University of Florida, as well as NC State with Max Scott, and Oklahoma State University have strong entomology programs and a pool of students that I think, if they're exposed to the work that's being done in Latin America, could really provide some broad-based training. Again, we just want to have an immersive model. If you want to think of this as phases, phase one, foundational training with our Latin American partners. Phase two, how can we operationally immerse them into either COPEG or other Latin American institutions? And then think about the long-term impacts. Again, we would like to begin their training at COPEG, but then fan out from there to other Latin American institutions that do work in screwworms. So the framework is this phase includes training so students can get an orientation to foreign animal disease response, which New World Screwworm are a part of, and then the initial technical instruction that they need just to do surveillance and to work on New World Screwworm. Phase two is really about everything, is being exposed to all the way from quality assurance and quality control within mass rearing operations to research and field surveillance and piloting new monitoring techniques. And then that last part that's critical to any kind of capacity building is how do we train them to engage with the stakeholders, whether it's governmental stakeholders or stakeholders on the ground, like such as livestock producers. Again, long-term impacts is that we're hoping after 2 years is that this is a model that can form a pool of candidates that can interchange both from U.S. institutions to Latin American institutions. And we really fully expect this to be a two-way street, that if there's Latin American institutions that want to partner with U.S. institutions, we want to provide that framework so they can do that. And again, it's just an immersive experience that we really need to get more boots on the ground and more people trained in this area. Again, needed input, and this can be, we can wait for this in the open discussion, is there capacity to train students in multiple institutions in Latin America? And I think there is, and I think FONTAGRO kind of provides a framework for that. Does the knowledge gap for New World Screwworm only exist in the US because we haven't dealt with it? Again, I think our Latin American partners and colleagues have dealt with this and can provide valuable experiences for students. And again, sorry for the typo there, but how do we identify Latin American institutions that can train students? And then Lastly, how can U.S. institutions help train students from Latin America? And so with that, that's kind of wanted a brief overview, just real quickly. Hopefully we didn't put us over too much time, but I just want to thank you for the opportunity.
Thank you so much, Justin. I think that was really valuable, and I think that the Screwworm is going to be on for a while, and so making sure we're training the capacity and building up our workforce to deal with it here and abroad is going to be really critical. And now I'm going to turn it over to Jasmine to moderate panel 3.
Great, thank you, Andrew. It is my pleasure to welcome you to our third panel of the day, which is on-farm control measures and private sector involvement. My name is Jasmine Bruno, and I'm a scientific program director at the Foundation for Food and Agriculture Research, or FAR, and our mandate is to facilitate public-private partnerships to support ag research. So our third panel that we're going to have today is really focused on the practical side of New World Screwworm, strategies that producers can implement on the ground, how treatment protocols are evolving, and then where private sector innovation can really fill gaps. I'm excited to introduce a fabulous group of panelists we have today that are going to share experience from research and practice. Our first panelist is Dr. Philip Kaufman. He is a professor and the chair of the Department of Entomology at Texas A&M University. And Dr. Kaufman, I'm going to pass it off to you.
All right, thank you very much, Jasmine. You all can see the screen, I assume?
We can see your side slides though.
Move here.
Yep, perfect.
So, I'm going to deliver the first part of this, and Ted Burgess at the University of Florida, got cut off at the bottom of the slide here, will be speaking toward the end. So the focus that we were provided was to discuss practical strategies for producers, treatment protocols, resistance management, development of new treatments, extension, and private sector engagement. And so it's been mentioned earlier on this webinar that management of screwworm really is integrated pest management. So using multiple tactics to address a larger issue, and that sterile insect technique alone will not work. It really requires participation by the producers and the public to help reduce the population of those flies as much as possible, so that the sterile insects do have an opportunity to do what we spend all this money on in rearing them, right? So again, our focus in the U.S. is that eradication is the goal, and we understand that that process will take some time. I'm telling my producers that this will be an issue for them for probably 10 years or more. And our Department of Agriculture a couple of weeks ago announced that they are investing in a new production plant to add to the production that is currently coming out of the Copeg plant. The announcement was for 300 million sterile flies per week, and that process, the construction of that facility and ramp up of production, is likely to take 3 to 4 years. And we are going to have to find alternatives up until that point. And I'm sorry for the image on the right, but this is an image of one of the key deer that was attacked when the screwworm arrived in the Big Pine Key off the southern tip of Florida 10 or so years ago. So again, as I mentioned earlier, reporting is critical. As I stressed to our Texas livestock producers that, we can only drop flies on your ranch if we know that your ranch has the fly, and that they're used to having their properties quarantined for fever ticks. But as I point out, to these ticks don't have wings, thankfully, and that this fly really doesn't care what a border is. I will add to one of the things that Dan had mentioned earlier about the Argentina-Uruguay border, is that screwworms are really not known to cross large bodies of water. And so knowing that that river system is there can be leveraged in any sort of an eradication program that might occur in some regions of Latin America. So when we look at management, what we have learned, and again, we're learning quite a bit because none of our producers have had to deal with screwworm. Many of the current producers are my age or older, and they were children when screwworm was last in Texas. And they tell me stories of what they remember, but again, it's memories of childhood. And so we rely very much on our Latin American friends to share with us how it is managed, where this fly has been present while we have been screwworm free. And what we've learned is that well-managed operations have far fewer issues. And so it comes down to operations that are able to and do routinely inspect their animals. They maintain facilities maintenance such that they prevent injuries, so there's no broken pipes with sharp objects that cause injuries. And then having a production practices that include ectoparasite management, such as for ticks. So this fly, as many of you are aware, is capable of laying eggs in wounds as small as a tick bite. And so operations that manage for ticks, say with insecticide containing ear tags, are known to have fewer screwworm cases. And again, it's not find it, treat it once and ignore it. You have to bring those animals in where you can monitor them and make sure that we don't treat a wound and not check on that animal during the healing process to facilitate an additional strike by the screwworm. And as we well know, the umbilicus of all newborns is very prone to strike by this fly. And so practices that treat the umbilicus of all newborns will certainly hasten drying and healing of that area, will prevent future infestations. So I'm going to leave a lot of the pesticide component to Ted to talk about. But one of the challenges we've had in the US, and we're currently facing it, is that for our insecticide producing companies to register a product in the US, they have to show efficacy data against a pest for it to be listed. Well, we didn't have screwworm in this country for the last 50 years or so. And so all of our products that were developed from the late 1970s coming forward have not had they've not had the ability to test it against screwworm. And so even though we have products that will very likely kill screwworms, those labels had not included screwworm on them. Therefore, it's technically illegal to use some of these products on your animals for screwworm infestations. We are working with our federal legislators or agencies to change some of this wording so that these companies are allowed to add these products, particularly if they have data from endemic countries such as those in South America, to add those. So right now we have essentially pyrethroids and an old organophosphate called Coumaphos, which has maintained that label, but it's a very old product and one that we'd rather not have to use because of some of the other challenges with using organophosphate products. So one of the things, and we've done a lot of training that goes into our extension programs, is to retrain our livestock producers and wildlife management folks on what to look for. So, screwworm is a very painful infestation, and so animals will express an irritated behavior. Oftentimes when the irritation is up on their head, you'll see excessive head shaking. When you get near these animals, they have a smell of decay, but that animal is not dead. And it's a very distinctive smell for those who have unfortunately had to smell that. And then again, seeing fly larvae or maggots in the wounds of living animals creates an automatic reporting process in the United States. So we direct our producers to bring in a veterinarian to remove all of those larvae, but those larvae have to be submitted through an official process where we send them to a single lab as part of our USDA APHIS group called the National Veterinary Services Laboratory. It's up in Iowa, where they have taxonomists that will confirm that it is screwworm or not. And even though many of us are capable of identifying those maggots, they do in fact have to go to this particular place. All right, so again, monitoring wounds is important to keeping populations low. The idea here is we've asked our producers to develop farm management plans. So all of our farms have or should have management plans for we'll call disasters. So if we have a hurricane coming in off of the Gulf, those producers pull out a plan that says move cattle from pasture X and Y up to pasture A, which is perhaps higher ground, 3 days before the storm is set to hit. Well, we've been asking them to develop similar plans for what do you do if screwworm arrives, so that they're very able to actively participate in protecting their herds. These include developing plans for different activities. If you're going to dehorn cattle, which is something some of our producers still do, then you need to have a plan in place for that, because dehorning creates a wound in a very, very sensitive area of the animals where the screwworm can rapidly gain access to the cranial cavity, which rapidly results in death. Castration, birthing seasons. We even have producers, so we have the opportunity here to shift our breeding season so that calves are born either late in the fall or during the winter when screwworm is suppressed because of the colder temperatures we have. Obviously, that's not something that's available to many parts of South America, but it is something where you can constrain your birthing season so that you can focus on those young and very vulnerable animals. And how do those protocols line up with the procedures you need to be taking? Training your employees, right? So you can have the best laid plans, but if your employees aren't informed as to what they need to be doing, as well as the importance for that, it's not going to result in a very successful plan. And then providing direct assignments for employees who will be held responsible for the animal's health, right? You can name an individual whose job it is to do these animal health checks and be responsive to any of these issues. And that may be a person that reports directly to the ranch foreperson, the ranch manager. And then one of the things is to be able to capture those animals. Are your animals approachable and accessible for evaluation, or are they in a silvopasture where the animals are partially in forested areas or swampy areas where maybe you can't get access to them? How are you going to handle those sorts of animal check issues? And do you need to move animals to more accessible areas during, say, a birthing season so you have access to them? So we encourage folks to be proactive rather than reactive. So what types of activities can be avoided that lead to the wounds, right? So facility maintenance, I mentioned. If you use herding dogs, the case that occurred in north of Veracruz, Mexico, was a young calf, I should say, that had been bitten by one of the ranch dogs, created the wound. And so that wound then led to the screwworm infestation. How are your animals able to be handled, and do you have issues with the bulls that you're using? And that can come into birth weights and whatnot that may create more wounds on your cows. Right? One of the things that's been suggested, and again, it comes down to what the local regulations are in your particular country, but can animals be pre-treated with a systemic insecticide to prevent infestations, such as we're going to castrate animals? Is that also a good time to treat them with a systemic, longer acting product that may provide protection to those animals during the period when they're healing from the castration treatment or dehorning event? Do you have facilities that are capable of holding your animals and allowing treatment of them? So squeeze chutes or compression areas. Is your animal are the animals on your ranch, is their disposition or their behavior such that they can be worked without creating further injuries? If you don't have a relationship with a veterinarian, we suggest that you make sure you do have one so that they are immediately on call if something should happen, that you need their assistance and you're not waiting several days for them to come to your ranch. And do you have the products that you need to treat the animals with on hand, and do you have a supply of those? So it may be some of those cans of permethrin that you may be able to treat a wound with, but if you have to stop and run to town to go buy those products, that's delaying and creating more stress for your animals, and those delays in treatment can be fatal. So make sure you have the supplies of the products on hand and resupply when those products are getting low. The other thing we suggest is, again, create a notebook with resources that are available to your staff. So whether those are fact sheets that are coming from universities and whether they're coming from the US or not, or those from your own countries, have those fact sheets and other informationals available on site such that they're easy to access and perhaps they're portable to take to the field. I'm going to turn it over here in a second to Ted. But again, come up with treatment protocols. You need to understand the rules and regulations of your country, as well as planning for resistance and resistance management. Resistance does happen. There's very few arthropods on this planet that haven't developed resistance to the products we use against them. But again, recognizing what are the available product classes and categories, what are registered and approved for screwworm. You know, we have to worry about here in the US. We have other products that are labeled for livestock, but not for screwworm, and recognize that if your animals are destined for export, making sure you're not using products that are not labeled for use in the destination countries where your animals are sent, such as, there are products that are available in South America that have no U.S. food animal approval, and if they're treated with those products and discovered, it can create trade challenges. So here are some of the things that we share with our producers are with suspected cases, take immediate action, right? So contact authorities. So in Texas, we have 2 groups that are helping facilitate responses should the fly show up. We have the Texas Animal Health Commission, which deals with our livestock, which are horses, sheep, goats, cattle, bison, and they also handle companion animals, so cats and dogs. We also have an agency called Texas Parks and Wildlife Department, so they handle all of our native wildlife species and in some extent our exotics. So Texas has a long history of bringing in wildlife from other countries and other continents. So we have thriving populations of we'll call them feral or wild populations of nilgai antelope and axis deer that are from the Indian subcontinent. We also have many other exotic species of wildlife, and Texas Parks and Wildlife are charged with those. Again, as I said, notify your private veterinarian such that if a wound is so large that you need assistance with it, you can get that person there rapidly. And do not delay in reporting. If your country or region has reporting requirements, letting folks know that the flies are active in that area can help minimize long-term consequences. And then in the US, we have our extension service, which in Texas it runs out of Texas A&M AgriLife Extension. We have county extension agents located in every county in Texas, and those agents are on call. They have access to greater networks and resources than an individual does. And as I mentioned, it's a law in the US that all myiasis or when maggots or larvae are infesting a living animal is called myiasis. In that case, all maggots from those animals are required to be submitted to the USDA National Veterinary Service Laboratory in Ames, Iowa. And as I mentioned earlier, Amarillo is a city up in the Panhandle area, up in this area of Texas, where all of our feedlots and large dairy farms are. Well, that's about the distance down to Brownsville in the southern tip of Texas. That's about the distance we have from where the cases are in southern Mexico. And so we have regional offices for our Animal Health Commission, and our producers are directed to contact those regional offices so that we can rapidly respond to any known cases in Texas. And lastly, one of our charges was to speak toward extension. As I mentioned, we have county agents in every part of Texas. We also have, we call them program specialists or extension faculty, located in many of our research centers scattered around Texas. And we develop fact sheets and other materials. So on here, you're welcome to take a photo of this if it would be helpful for you. If you type into Google Texas A&M or TAMU screwworm, you'll get rerouted to our AgriLife Extension website, which is here. And on that website, there's a link to New World Screwworm. And as we develop new materials, we're posting them on this site. We also do trainings across the state for our veterinarians and for our extension folks, as well as for the public. We've held several of these in South Texas already and have drawn crowds in the hundreds to these, as people really are needing to be updated on what our screwworm issues are and bringing them up to speed. I had put this slide up earlier, and welcome to come back to it during the discussion. As Ted will talk about, we need research and assistance in development of new systemic longer-acting insecticides, medium-term priorities, developing perhaps a lethal bait system that is environmentally sustainable and New World Screwworm specific. There was one developed back in the 1970s, and it was essentially small discs of a product that were dispersed from airplanes. And the intent was to bring flies in an area that would be attracted to this bait, feed on the bait that contained an insecticide, and kill the adult flies. Although there have been a couple of publications on that research, unfortunately, the research wasn't done up to what we would consider modern scientific standards. And so the results of that, where they claim 90% suppression, are somewhat subjective. And that doesn't mean that this isn't an approach that could be developed and be useful, but the efficacy is likely not to be 90%. But again, using an approach like this could be beneficial if we develop it correctly. And some of the short-term actions are, again, for country-specific needs, or develop protocols for action, develop production action items for common procedures. So if you're going to castrate, here are the things you need to do. Develop lists of appropriate pesticides, and encourage control of other blood-feeding arthropods like ticks or even some of the biting flies to reduce wounds on animals. Working to develop best management practices for different types of livestock, and engaging with federal agencies on training for staff. And with that, I will stop sharing my screen, and I believe, Ted, you are up next.
All right, let's see here. All right, everyone can see that all right? I'm excited to be here. My name is Ted Burgess. I'm an assistant professor of veterinary entomology at the University of Florida, and I study ectoparasites of livestock importance. And I've really specialized in flies, house flies, horn flies, similar types of flies to the New World Screwworm for pretty much my entire career. So I wanted to initiate, what I'm about to say. This little highlight at the bottom here about the little yellow highlight is indicating knowledge gaps in each of the slides. So as you're looking through these slides, make sure your eye gets trained on these yellow highlights, and then I'll sum these up towards the end. So we're going to start with how is New World Screwworm managed with chemicals? As Dr. Kaufman mentioned, sterile insect technique is the way that it can be eradicated, but management of wounds and management of isolated outbreaks of it are going to be primarily done with chemical control. So this is a crucial part of the toolkit that is needed to manage New World Screwworm. And so the types of products that are available, to the best of what I could find in Latin America, are products that primarily target the adult life stage and then the larval life stage. We know that this fly has 4 life stages: the adult, the eggs, the larvae, and the pupae. We know that the females will seek out wounds to lay their eggs on or near those wounds, or in other cases, mucosal membranes. And then the larvae are what do the damage to the animal feeding on live tissue. And so the application types are typically categorized in 3 areas, and these kill either the larvae and or the adults. And this is going to be wound management in the case of larvae, prophylaxis of infestation or female landing, and then ancillary effects like tick control. So applying products that are going to control other types of parasites, in addition to things like dewormers for things like stomach worms. So what I'm going to talk about today are 2 things. We're going to talk about resistance and what we know about resistance in New World Screwworm, and then a little bit about novel chemistry discovery, or how we can utilize chemistries that are either existing or do not yet exist, but kind of the pipeline that we can use to explore those. And I should also qualify this statement too. One of the main aspects of my research program is in the toxicology of insecticides and the discovery of novel chemistries, and then also resistance management, including characterization of resistance mechanisms in veterinary pests. So this slide is kind of a pretext to all of the stuff I'm about to talk about with resistance. So some facts to get straight here. The first being that not all mechanisms are going to be found in resistant arthropods. And some mechanisms are going to manifest stronger phenotypes, and this is often species dependent. A good example of this would be certain target site mutations in mosquitoes. Some have been shown to confer very strong resistance phenotypes, while those similar types of mutations in other species of mosquitoes, those target site mutations confer less intense resistance phenotypes. So I like to explain this with this triangle here. And we start with absorption and distribution, and that's going to be defined as a reduced cuticular or gut penetration, or spread of the chemicals across the contacted surface. It's one of the important aspects of how an insecticide penetrates through the cuticle, is how it distributes across the cuticle. And so the triangle then starts to broaden. So we know that reduced cuticular penetration is not generally considered in the literature across all relevant arthropods to be a particularly intense resistance mechanism. But then when you compound that with other resistance mechanisms, the next one would be metabolism and or excretion, which we define as the ability to break chemicals down quickly or more efficiently into less toxic metabolites, and or how they excrete those chemicals, either as the whole chemical or as the metabolites. There are enzymatic processes in arthropods that enable this to occur. And then the third one would be target site modifications. These are usually amino acid substitutions in the target site. Some examples for this are KDR or RDL mutations in the ion channels or the receptor sites for ligand-gated ion channels in the nervous system of the arthropod. And a lot of the chemicals and in fact, all of the ones registered, or at least the vast majority of the ones registered for New World Screwworm in Latin America fit into this neurotoxic category. And what those amino acids do is they change at a key location where the insecticide will bind to that ion channel or that ligand-gated channel. And it reduces the ability for that chemical to bind to that channel and have its toxic effect on the channel. And when you compound all of these things, as you add more of these mechanisms, you broaden the base of the resistance phenotype. So the arthropod will become more and more resistant. And again, not all of these mechanisms are present in every resistant arthropod. So that's a really key thing. And then behavior would be kind of a fourth one that's kind of a Pandora's box on its own. This is the behavioral avoidance of chemicals. And there's a lot of stuff that kind of drives that phenotype. Resistance is always possible. I interface with industry a lot, and we always hear the next new chemical coming down the line is resistance proof. That's just not a realistic way to look at pesticides. There's always going to be a mechanism for resistance. And some of these resistance comes about in several ways. Commonly, it's going to be subtherapeutic exposure. So if you think about things like a poron, the site of application is going to have the highest residues, but as it gets farther away from the site of application, those residues are going to reduce. And in certain parts of the body, those residues could become subtherapeutic. There's continuous exposure. Larvae are going to be the prime example here for New World Screwworm. And this is when the insect is just being constantly exposed to that. So think about things like systemic control solutions like injectables like ivermectin or doramectin. Overuse of a single chemical is also going to do this. It's going to give the opportunity for that population to be selected for those resistance mechanisms. And then high generation turnover is also a key factor for the evolution of resistance over a relatively short period of time. And we know in the New World Screwworm, they are a highly fecund organism. They lay a lot of eggs and can get out of control really, really quickly. And so the first knowledge gap here is there is a limited knowledge that exists on these mechanisms in New World Screwworm. So before I go into what we know about resistance in New World Screwworm, I'm going to talk a little bit about the chemicals available, and Dr. Kaufman went into some stuff about this. And by no means is this list exhaustive. And this is actually one of the knowledge gaps I will get to a little bit later. But we can categorize kind of the most commonly used pesticides for the management of New World Screwworm. We, of course, have organophosphates and carbamates, things like coumaphos and propoxer, and these are going to be in dust, smears, porons. And I want to introduce this idea to anybody in the audience right now who is not aware of IRAC. So IRAC is the Insecticide Resistance Action Committee, and this is the group that categorizes insecticides. And they're very involved in the management of insecticide resistance by categorizing insecticides into common groups with common modes of action. And as I alluded to in the previous slide, single uses of single chemicals or even classes of those chemicals can help to drive resistance. So an important part of management for resistance is the rotation of these chemicals to try to slow down the evolution of resistance in these populations. So organophosphates are going to be acetylcholinesterase inhibitors. And these, of course, have problems with use in animals and human exposures. And it's one of the subjects in question for how the EPA registers new insecticides. Then there are phenylpyrazoles. Fipronil would be an example of this. There are porons available with fipronil in them. These are GABA-gated chloride channel blockers. So this is going to be a ligand-gated channel, GABA being the neurotransmitter that reduces excitability of nerves. And then pyrethroids, permethrin is a good example. Sprays and porons are types of products available with these types of compounds in them. These are sodium channel modulators. Ivermectins are going to be ivermectin, doramectin. These are going to be injectables or even porons. And these are glutamate-gated chloride channel allosteric modulators. Glutamate is the main neurotransmitter at the neuromuscular junction of arthropods that allows electrical signals to be transmitted to the muscles to allow normal muscular movement. And then isoxazolines, which is a relatively newer class of pesticides from the other ones on the list. Fluorolaner is an example, and there's a product available called Exalt. That is a poron, and this is a GABA-gated chloride channel allosteric modulator. And it is notable because some of the target site mutations that are common with these chloride channel target chemistries, the isoxazolines bind to a unique place in that channel away from where that target site mutation is. And so those resistance mechanisms that confer some resistance to phenylpyrazoles or ivermectins do not have an effect when isoxazolines are used. I've also put up here the aphids, USDA APHIS list of products that are recommended that have some kind of language for screwworm on them. These are QR codes that will take you directly to the websites. And definitely avail yourself of that information. So these products come from a combination of registrations. There's extra label use. And then ancillary effects, like we talked about previously. This is things from control of other parasites like ticks and stomach worms. So what do we know about resistance in New World screwworm? The answer to that is not a lot, really. At the very end of this slide deck is the citations table. So if you're interested in looking deeper into some of the citations from this table, please go to that last slide, and you'll be able to find all of the DOI numbers and the citations. And it's interesting to read. For organophosphates, these are kind of historically been heavily used. There, of course, is documented resistance to organophosphates, carbamates. A mechanism has been proposed. The main mechanism for that is a metabolic mechanism. It's been proposed an esterase called esterase E3. There's a mutation in that esterase that allows for hydrolysis to happen of organophosphates and carbamates. The issue with this literature is that there haven't been any the typical standard tests that are done to infer or confirm these mechanisms do not exist in any of these citations. They heavily rely on past literature of closely related flies, and they use that basically to infer what is causing the resistance to organophosphates in New World screwworm. So that's an important thing too to bring up. So confirmation, and I've given a list here, and you know, this list can serve as kind of some actionable items for working towards a better understanding of resistance in New World screwworm. Phenylpyrazoles, so this is going to be fipronil. There is no documented resistance, no mechanisms proposed, and in fact, no citations on that, that class. Pyrethroids, there are a couple of papers on that. Again, they tie it to esterase E3, which again, there have been some papers in other flies to show that that is a mechanism of metabolism. However, in these citations, again, they do not use any of those kind of standard tests to confirm that mechanism is the main mechanism of resistance in New World screwworm. Avermectins, there is documented resistance, but a mechanism hasn't been proposed, and it also has not been confirmed. And then isoxazolines are new enough with really limited documentation of how that product is being used across Latin America right now. So there is no documented resistance, and there really has been, because of that, no mechanism proposed, and certainly no citations. So I want to go into this list of kind of conformational studies that are used in toxicology to confirm a mechanism is causing resistance. And again, these studies do not do any of these mechanisms. And I think that this is an incredibly important knowledge gap that needs to be addressed if we're going to start to understand how to manage resistance. So these include reduced penetration kinetics through the cuticle. This is going to be the absorption distribution resistance mechanism I talked about earlier. Expressing the detoxification enzyme in an insect cell line or some other kind of vectored cell line, and then doing metabolism studies on this. This is how the basis for that esterase E3 story came about in other flies, and it was confirmed in those other flies with this type of study. But this type of study is absent in all the New World screwworm literature. Synergist dose response assays are another very simple one that we rely on to imply an enzyme class as a potential metabolic detoxification mechanism for resistance. And again, these studies do not have those types of assays in them. There are classical crossing experiments that can be done where you can identify a putative mutation in a target site. You can selectively cross ones with and without these mutations, and then you can do dose or concentration response assays on them to start to get towards or narrow down that target site mutation may be having an effect on that toxicological phenotype. And then kind of a gold standard for this is electrophysiological evidence. There are ways to express these ion or ligand-gated channels in oocytes. There are in situ electrophysiological methods, certain types of voltage and other types of patch clamping methods where you can really isolate out the physiology of the channel and determine if the channels with those mutations have altered properties when they are exposed to the pesticide. And then for organophosphates and carbamates only because they target the acetylcholinesterase enzyme in the synapse of the nervous system, in synapses of the nervous system. That enzyme actually has a really nice system where you can use a model substrate to determine how much of that model substrate acetylcholinesterase will process in the presence of concentrations of organophosphates or carbamates. And again, those studies are, those types of studies are absent in the literature right now. What the literature primarily has in it is it ties resistance to this esterase E3 mechanism, and then they rely on building on that story with continual molecular surveillance of the mutations in that enzyme. And so it's really important that we actually look at that in the real organism. And I go back to my previous slide about talking about resistance mechanisms, in that not all organisms show all resistant mechanisms, and those resistance mechanisms are highly species dependent. So just because this was seen in a closely related fly does not mean that it necessarily has the same kind of effect in New World Screwworm. And so we really need to back up and be able to do some of those studies, so those conformational studies, so we really know what's going on. So one way we can manage resistance, besides a better understanding of the mechanisms and more in-depth confirmatory studies of those mechanisms, is the development of novel chemistries. So this list right here is, these are IRAC chemical groups. These are not currently used in New World Screwworm management, or there are papers that have tested some of them for efficacy. And these are all on the EPA reduced risk, and they all have distinctions in the EPA reduced risk category that say they can be used for animal feed or food exposure. And so while that's no guarantee that they can be used on animals, for a number of reasons, they at least narrow down a list of potential candidates that we could start to explore, that may give us some different chemicals in the toolbox. And these are unique because they have different modes of action to the stuff that is currently being used. So things like spinosins, these are nicotinic acetylcholine receptor allosteric modulators. Bacillus thuringiensis toxins, these are microbial disruptors of the insect midgut membranes. Oxadiazines like indoxacarb, this is a sodium channel blocker. It must be activated inside the insect's gut. It actually hydrolyzes a part of the structure to activate it inside the insect's gut. Diamides like chlorantraniliprole, this is basically affecting the electrical system in the muscles that allow muscles to contract, and it essentially paralyzes the insect. There are also other isoxazolines as well, like afoxolaner and ceralaner, and there's some literature from work we've done, actually, from grant funding, that shows when resistance develops to one isoxazoline, it does not necessarily confer resistance to other isoxazolines. And again, this is in a closely related species, so there's no guarantee that that's how it will work in New World Screwworm, but at least gives us some promising information to base development efforts off of. Some other areas to think about. It's reimagining old chemical classes. Something that I think would be worth looking at is there has been about a decade long, maybe a little longer than that, push for the species selective organophosphates and carbamates. So the way that those molecules bind to acetylcholinesterase, there are some very specific residues on amino acid residues in the acetylcholinesterase protein that can be very species dependent, and mammals tend to have a different complement of amino acids in those binding sites than arthropods do. And even within orders and families of arthropods, we find differences in those amino acids in that binding site. So that would allow us to develop chemistries that can bind selectively to species-specific organisms. And there are some great papers out there on this, things in aphids. There's another great paper, Swale et al., 2013. I bring this one up explicitly because although this one was looking at control for cattle fever ticks and sandflies, one of the key components of this study was these species-selective anticholinesterases or carbamates that they were working on. They tested acetylcholinesterase extracted from bovine brain. And they found that a number of those lead chemicals that they were developing had very, very, very low binding to bovine acetylcholinesterase relative to the arthropod that they were trying to target, acetylcholinesterase, sometimes in the order of 200 to 300-fold lower binding affinity to the bovine acetylcholinesterase. So I think that this gives us a good kind of idea that we can reimagine old chemical classes for more species selective chemistries that we can use that would not endanger our animals or our producers that are having to handle these chemicals and control control the New World screwworm. The other notable thing in Swale et al. 2013 is that a lot of those lead compounds that they were testing, they tested in acetylcholinesterase that had target site mutations in them, and they found that a lot of them overcame that resistance mechanism. So this is further evidence that we can really tighten down this chemical class potentially for use against New World screwworm. And then, of course, there's just the kind of broad exploration of new modes of action as well. We're always needing new modes of action for chemical rotations to manage the evolution of resistance.
Ted, sorry to interrupt you. I just wanted to, we're getting really short on time.
Sure.
We have one more panelist in this panel. I just wanted to okay, thanks.
I have two more slides, yep. So some barriers to novel chemistry development. We're going to have to deal with withdrawal times. This is going to be important. Dr. Kaufman talked about this earlier. The animals have to clear the chemical from their body before they can be put into the food chain. And volatile, violative residues are evaluated in commercially processed animals. So this can be a barrier to novel chemistry development. Non-target effects, of course, we don't want to kill things outside of the pest. Environmental persistence is going to be, a whole battery of data that's going to need to be generated in order to make these chemistries viable. And then my last slide here are going to be some of the research priorities. And we can go, when we start the discussion, we can go back to this slide. And then the conclusions here are kind of going over some of those yellow highlighted areas that I mentioned in the earlier slides. Here's the table for anyone.
Oh, apologies. Great. Thank you, Dr. Burgess and Dr. Kaufman, for those fabulous presentations. We have one more panelist, Alejandra Diaz, who is an international specialist in agricultural health and food safety at ILCA. So it's a pleasure to welcome Alejandra.
Saludos a todos. En este momento voy a hacer la presentación. Espero que tengan oportunidad ya de ver la presentación. ¿Me confirmas, Yasmin?
Sí, se ve.
Yes, we can. Thank you.
Muchas gracias. Bien, en principio, pues, como nos señaló Yasmin, trabajo en el IICA, en el programa de Sanidad Agropecuaria, Inocuidad y Calidad de los Agroalimentos. Y particularmente estoy desde el 2024 colaborando con la coordinación regional en un plan de trabajo que tenemos el IICA y EIFIS-USDA en los trabajos de capacitación y comunicación para el gusano barrenador de ganado. Entonces voy a compartirles a ustedes básicamente muchos de los aprendizajes y acciones que estamos nosotros desarrollando a la fecha con Centroamérica y México. Bien, para nosotros, pues, incluso desde la perspectiva misma de este plan de trabajo, nos abocamos en capacitación y comunicación. Consideramos que es un elemento importante, un elemento integral, que es más que un complemento. a los aspectos de prevención, de control y de erradicación del gusano barrenador del ganado. Como punto de partida en nuestro trabajo, entendíamos que tenemos que, pues, tantos años de estar erradicada esta enfermedad, consideramos importante tener una base de conocimiento. Y en esa base de conocimiento implicaba tener y construir una red regional. Para ello, levantamos con los diferentes países esta red a través de responsables de capacitación y comunicación y arrancamos inicialmente con el sector de agricultura, que es con quien directamente trabaja el ICA, ¿no? Y para ello, y aun cuando todavía no se había extendido en el mediados del 2024 el gusano barrenador en toda la región centroamericana, pues era importante que en una primera instancia todos conociéramos de qué estábamos hablando. Para ello, nuestra primera visita y encuentro fue precisamente en Panamá y fuimos a las instalaciones de COPEC. De esta manera, era importante que cada uno de los responsables, estamos hablando del sector de agricultura, tanto de capacitación y de comunicación, conociera realmente, directamente al gusano barrenador del ganado, para que no quedara duda. Incluso un olor característico que tiene el gusano barrenador y que, pues, tras conocerlo, realmente queda tan fijo en la memoria que después de sentir esa miasis, nunca uno se olvida. Y precisamente con ellos empezamos a trabajar una serie de acciones. Nuestras acciones, si bien son varias las audiencias, y en este panel vamos a concentrarnos en el tema del productor. Nuestras acciones corren desde capacitación, que significa presencialidad, virtualidad, como el que estamos desarrollando ahora, para llegar ahora últimamente ya en la formación de capacitadores. Acudimos a recursos de información y comunicación. Esto implica desde materiales más simples, infográficos, trípticos, vallas publicitarias incluso para las campañas de comunicación, y estar presentes en ferias ganaderas, en congresos o cualquier evento donde vamos a encontrar una participación mayoritaria de nuestra audiencia específica, en este caso como los productores. También recurrimos a todos los medios, redes sociales y medios digitales. Esto implica que en algunos casos acudimos, por ejemplo, al perifoneo, utilizando guiones consensuados, siempre desde la sede central. Procuramos desarrollar unos máster que luego vienen siendo ajustados, adaptados, aprobados por los propios países, por las autoridades nacionales y después difundidos. Entonces, ese es el mecanismo de trabajo que nosotros llevamos. Entonces, a nivel del productor, hemos entrado en una primera parte, digamos así, a un nivel de sensibilización. Inicialmente ha sido un trabajo muy fuerte de sensibilización, con mensajes claves. Para ello identificamos cuáles tenían que ser esos mensajes, de dónde íbamos a partir, pero inclusive el trabajar en campo implica atender a sus familias y particularmente a la escolaridad, a los jóvenes, y por eso que entramos también a trabajar en una serie de charlas de sensibilización a través de escuelas y también colegios técnicos agropecuarios. Y como muestra esta imagen, también participantes con stands en diferentes congresos o eventos divulgativos donde aprovechamos hacer toda esta, digamos, divulgación. De diferentes maneras, actuamos también en ferias, en días de campo, que consideramos que estas últimas acciones, que son los días de campo, donde mezclamos presentaciones y también actividades específicas y demostrativas, cobran mayor importancia y hay una necesidad muy amplia a nivel del sector productor. Entonces, tenemos desde grupos pequeños hasta grupos masivos, muy amplios, donde venimos trabajando a través de los países. Este, por ejemplo, es la muestra de días de campo que se realiza en esto, un trabajo muy amplio y muy coordinado con las diferentes autoridades sanitarias en los países y un socio muy importante como es COPEG. Entonces, en sesiones demostrativas, donde al aplicar alguna práctica como el descorne, se hace pues una curación de las heridas de manera apropiada para que todos los productores participantes puedan estar vívidamente conociendo la práctica a desarrollar y de una manera adecuada. Creemos que estos son elementos que tenemos que seguir reforzando y que debe ir a la par de cualquier otra práctica en ese enfoque integrado que nosotros también propiciamos. Pues bien, a esa etapa de sensibilización con el que hemos corrido muy fuertemente en los países, hemos entrado ahora a un proceso más estructurado en términos de formación. Entonces, esta es una guía de capacitador que hemos elaborado, hemos construido de una manera colectiva con los diferentes responsables de capacitación de los países, en muchos casos los mismos directores de los programas de gusano barrenador del ganado. Entonces, en esta guía del capacitador hemos concentrado los elementos clave que debería impartirse en los cursos de capacitación, de manera que estamos generando un mensaje no solamente clave, pero sino asegurándonos que se esté brindando de una manera estandarizada. Entonces, al productor le mencionamos que obviamente es muy importante el tema de la mosca, pero por ejemplo que hay que concentrarnos en los gusanos y nuestro enfoque está ahí respecto a evitarlos con la curación de las heridas. Entonces, en la primera semana de agosto tuvimos este encuentro con los que denominamos formadores regionales en una semana. Hemos tenido trabajos teóricos, mucho intercambio horizontal y finalizamos con un día de campo en un lugar del interior de Panamá para completar el trabajo de formación. Y tenemos ya incluso un programa de réplicas porque estos formadores regionales harán un papel de réplica en sus países, digamos, formando muchos más capacitadores. Entonces, al igual que esta guía, tenemos también un manual del participante, que es precisamente ya el que recibe el participante como tal. Entonces, algo interesante que hicimos aquí es que nosotros partimos de una concepción de que esto tiene que tener un enfoque de una salud. Por lo tanto, no nos quedamos exclusivamente con el sector de agricultura, sino que aquí también ampliamos al sector salud y dentro de nuestros formadores que aquí aparecen a nivel de los países Centroamérica y México, también está el sector salud, ¿no? Entonces, en coordinación precisamente con la Secretaría Ejecutiva del Consejo, el COMISCA, a nivel del SICA, nosotros establecimos las coordinaciones para que hubieran representantes de cada país en este evento. Y agregamos también a un participante del sector privado. Entonces, ¿por qué? Porque precisamente el protagonista importante en el campo pues es el productor. Entonces, la representatividad del sector privado está aquí presente y en conjunto formamos las réplicas que hasta diciembre de este año tenemos planificado, pero que también estaríamos trabajando el 2026. ¿Cuáles son los principales elementos que tenemos en esta formación? En primer lugar, pues partimos esto en 4 módulos. Entendemos que es una capacitación que iremos reforzando. Estamos pensando ya en algunos módulos específicos, especializados, pero hemos arrancado con uno que siempre ha sido el punto de partida en nuestros elementos, las charlas de sensibilización, que es conociendo al gusano barrenador del ganado. Ahí participamos, incluso con el tema del enfoque de una salud, con una serie de ejemplos, pero es tan importante que el productor conozca el ciclo del gusano barrenador del ganado, porque así va a entender cuáles son las razones por las que incorporamos ciertas metodologías o ciertas intervenciones, y porque es importante entonces que no haya herida que pueda traer a estas hembras a depositar sus huevos. También consideramos, y a nivel del productor, hay que hablarles con datos duros, hay que hablarle con elementos concretos, y esto implica el que conozcan los impactos. Entonces, aquí hemos hecho un gran esfuerzo de recolectar información ya existente respecto a los impactos económicos, aun cuando hay impactos en salud pública, impactos en la fauna silvestre. Sin embargo, creemos que todavía este es un aspecto en el que necesitamos seguir trabajando. Estamos basándonos en un reciente informe que sacó el Corfoga de Costa Rica, donde ellos hacen un análisis sobre el costo de los tratamientos y también, obviamente, ponen en pie el beneficio que significa un tratamiento oportuno. Esperamos pronto trabajar un análisis económico, una estimación aproximada sobre estos impactos también en los próximos meses. El otro elemento importante y hace parte de un módulo específico en esta guía de capacitador son las buenas prácticas pecuarias para la prevención y control, que engloba muchos de los elementos que también señalaron expositores anteriormente. Y cerramos esta primera, digamos, guía con un elemento que recoge y resume varios de los puntos que tratamos en módulos previos, pero con un elemento que es parte del eslogan de la campaña que tenemos nosotros a nivel de comunicación, es que sin heridas no hay gusaneras, sin heridas no hay gusano barrenador del ganado, donde hacemos hincapié de varios elementos y varios tratamientos que en su mayoría son bastante similares en nuestros países. Les mencionaba el tema del enfoque en la salud. Por eso que nosotros partimos con una campaña, porque hay que entender que hay una corresponsabilidad incluso de la misma sociedad. En la medida que esto afecta a los animales de sangre caliente, están también involucrados las mascotas, están también involucrados o posiblemente afectados. Ya lo vimos con los casos que se dan a nivel humano. Entonces, todo nuestro material comunicativo arranca y mantenemos ese eslogan de campaña, que juntos podemos contra este gusano barrenador del ganado. También transitamos a la importancia de revisar y de curar, esto directamente con los productores, para después, ahora, con más fuerza en el 2025, con sin heridas, no hay gusaneras, precisamente uno de los títulos de nuestros módulos de capacitación. Pues bien, dentro de las buenas prácticas pecuarias, yo no voy a ahondar sobre varios de esos elementos, esto es bien, muy conocido, pero particularmente un elemento importante son cuáles son las recomendaciones clave para los productores para que el diseño o sus instalaciones realmente sean tan apropiadas, tan seguras, que no se estén generando oportunidades para lesiones o para los riesgos a esas lesiones. El hecho de que tengan estructuras al que no han tenido cuidado y hay clavos expuestos, que haya bordes filosos, simplemente que haya cercas rotas, y la necesidad de que haya inspección, ellos hagan una reparación continua, de manera periódica de sus instalaciones, es muy importante. Aquí la visión es que tienen que reducir el riesgo de que esos espacios puedan convertirse pues, en puntos de entrada, en oportunidades de infestaciones de gusano barrenador del ganado. Otro elemento en esas buenas prácticas pecuarias es el entendimiento que todo lo que hagan ahí precisamente es para prevenir las heridas. insistimos y por eso que en todos nuestros elementos infográficos estamos incorporando que hay que evitar las heridas, porque sin heridas realmente no va a haber gusano marginador del ganado. Esta imagen lo hemos construido, a través del aporte que nos brindan en información las diferentes autoridades sanitarias de los países. Y en algunos otros casos lo hemos complementado con información que tomamos de la Organización Mundial de Salud Animal. Entonces, en particular, nosotros vemos que los animales o las especies más afectadas es el bovino, pero seguidamente también del canino. Está también porcino, equino y otros. Y en pequeñas cantidades se han visto casos también de animales silvestres, ¿no? Pero estos son elementos que nosotros compartimos con nuestro sector productor para también señalarles cuáles son esas principales afectaciones o dónde estaban esas lesiones donde se han producido esas infestaciones. Y en primer lugar encontramos a las heridas por golpes, por trauma, esos manejos seguros de las instalaciones, por eso vamos con eso. Pero también miren, va con algo que dice ombligos de recién nacido. Entonces están también los temas de mordeduras, de picaduras, de murciélagos. También tenemos el areteo. Por eso la importancia de que esos programas de trazabilidad vayan en conjunto de asignarle claramente el aleteo, pero sin el descuido de que toda herida tiene que llegar a una cicatrización completa. El marcaje también es parte de estas lesiones o los lugares donde se producen las infestaciones. Tenemos también el tema de las garrapatas. Entonces, por todo ello, consideramos que tiene que tener, además de ese enfoque integral que combina una serie de prácticas, es la importancia de trabajarlo intersectorialmente, porque esto nos hace también un llamado importante, que con la fauna silvestre todavía hay un trabajo, digamos, muy poco. En la prevención y manejo de heridas, pues nosotros compartimos a través de diferentes mecanismos los pasos esenciales para la prevención, como mencionamos, pero también con algunos refuerzos clave, ¿no? Es cierto que hay que evitar las heridas con ese buen manejo, con las prácticas seguras, el que se pueda hacer una detección temprana y particularmente también un tratamiento inmediato. Consideramos que esa sumatoria va a tener una prevención efectiva. También insistimos en la importancia del reporte, en la importancia de hacer la notificación. Cuando conversamos con los productores, algunos de ellos nos indican, si yo ya sé curar, ¿por qué voy a reportarlo? ¿Por qué voy a notificarlo nuevamente? También encontramos temores en algunos casos donde el productor teme hacer una notificación porque cree que su finca va a quedar cuarentenada o simplemente ese animal ya no lo va a poder comercializar. Entonces, hacemos este levantamiento de información porque a través de eso nosotros generamos información real que rompa mitos, que corrija esa información errónea a través de las campañas de comunicación que seguimos. Muy importante también, y acá señalarlo recientemente, en el uso de los medicamentos veterinarios. Entonces, las buenas prácticas también comprenden que además de ese tratamiento y de la prevención, pues tiene que haber un buen uso, de un uso responsable de esos medicamentos autorizados. En principio, y es lo que estamos sosteniendo por ahora, que es el compromiso de respetar la forma de uso, pero también respetar los periodos de retiro. Mucho más todavía cuando estamos hablando de animales lecheros o que se van hacia el sacrificio. Entonces, reiterarles con el tema del registro, reiterarles cómo están almacenando esos productos y que realmente tienen que hacer un uso correcto con equipos también calibrados. Entonces, consideramos que es todo un conjunto de recomendaciones, en algunos casos que son elementos obligatorios porque forman parte ya de no solo de buenas prácticas, sino de elementos regulatorios a nivel de los países. Complementamos con la importancia de las buenas prácticas en el transporte de animales, ¿no? Entendemos que, obviamente, que solo se deben transportar animales sanos, animales sin lesiones, y claro está, sin gusaneras, ¿no? Entonces, eso implica también tener adecuados vehículos de transporte, el que también tengan la seguridad de separar y observar los animales que están recién llegados, que adopten sus protocolos de bioseguridad, la importancia incluso de hacer una adecuada limpieza de transporte, porque los residuos orgánicos o el excremento de los animales también pueden servir pues de un lugar especial para que cuando caigan las pupas o salten los gusanos en ciertas fases, pues realmente puede ser propicio para que sigamos propagando el gusano barrenador del ganado. Siempre está de una manera transversal nuestro mensaje de que sin heridas realmente no vamos a tener gusaneras. El control del movimiento animal es sumamente importante, no solo por las condiciones del transporte, como señalamos, sino que pues esta es una enfermedad transfronteriza. Entonces, esto implica hacer más inspecciones sanitarias, no solo previo al traslado. Aquí tiene que jugar un rol importante los sistemas de trazabilidad bovina, los sistemas de trazabilidad en general, respecto a las autorizaciones o las guías de movilización oficiales, la prohibición para el transporte de animales heridos o infestados, y esto tiene que ser complementado con mucha capacitación sobre, por ejemplo, la detección temprana de síntomas, sobre el manejo adecuado del aislamiento y una aplicación segura de medicamentos. Aquí hay algo interesante y es algo que también lo está implementando México y está haciendo una cooperación horizontal a otros países. El uso de caninos, ¿no? Entrenados que puedan detectar con el olfato la miasis. Entendemos que esto en el sur es bastante utilizado, incluso en los propios rebaños, que parece ser que no requiere un entrenamiento pero tan costoso, pero que podría ayudar incluso en las fincas, ¿no? Tener animales entrenados que puedan identificar la miasis. Bueno, hay buenas prácticas adicionales también respecto a restringir el acceso a vehículos o personal externo a las fincas, el que puedan planificarse rutas seguras, tiempos adecuados, etc. Entonces, el movimiento controlado, entendemos que para nosotros que realmente va a proteger la salud del rebaño y va a frenar la propagación del gusano. Nosotros recopilamos para esta formación diferentes protocolos de los países y encontramos más o menos esta similitud y lo plasmamos en diferentes pasos, ¿no? Entre la aplicación de un tratamiento con larvicida, un paso 2, el que se retiren las larvas y los huevos, eso estamos hablando ya de los animales infestados, particularmente, y por eso lo tengo acá anotado en rojo, el explicarles a ellos que nunca se arrojan las larvas al suelo, ¿no? Porque ahí, nuevamente, pues vamos a encontrar que esta es una de las razones por las que se puede seguir propagando. Entonces, son aprendizajes.
Hola, Alejandra, disculpa la interrupción. Diría que quede un minuto nomás, por favor.
Ya voy cerrando con la última pista. Esto con la recolección de las larvas, algunas recomendaciones, y ya voy a finalizar con la última vista, las últimas digo. El intercambio de experiencias es muy importante. Aquí tenemos, por ejemplo, el sector privado, el presidente de la Federación Ganadera de México, compartiendo a diferentes participantes de la región, pero también a un público de Panamá. las diferentes materiales que acompañan a nuestras actividades de capacitación, que es a través de redes sociales, con mensajes que vayan a cubrir la desinformación y particularmente que indiquen exactamente qué es lo que se necesita. Como prioridades, consideramos que la formación y la comunicación tiene que ser continua. Debemos seguir con los simulacros. En México hemos hecho varios, pero tenemos que continuar con ellos. El intercambio de experiencias entre productores. Creemos que todavía hay necesidad de manuales específicos, una mayor participación del sector privado. Hay también la necesidad de ampliar la oferta y el acceso a medicamentos veterinarios registrados. Hay mucho interés del productor de preguntarnos cuál es el más eficaz, cuál es el más efectivo. Pero también esto nos invita a revisar nuestro programa de vigilancia de residuos de medicamentos veterinarios, porque estamos viendo claramente que hay un mayor uso de ciertas sustancias que deberían ser parte de esos programas de vigilancia. Creemos que hay mucho trabajo por hacer el manejo de resistencias, lo dijo anteriormente el expositor. Hay necesidad de nuevos tratamientos, el desarrollo de estudios de impacto, inversiones en fincas. Esto probablemente implica generar incentivos, programas, incluso con seguros agropecuarios, pero se requiere hacer un arreglo en una finca, el mejora de las cercas, eso requiere inversión, ¿no? Entonces, y obviamente trabajar con el sector privado al respecto. El control de movimientos de animales, es muy importante seguir reforzándolos y hacer más uso de la inteligencia artificial, por ejemplo, para control en animales en fincas, tal vez colocando cámaras en esos lugares de confinamiento para detectar temperaturas o detectar que el animal no se mueve y así poder ir a controlar, o el uso de drones. Entonces consideramos que podemos discutir mucho más acerca de todas esas oportunidades de inversión que puede haber a nivel público-privado o privado para los aspectos de innovación. Muchas gracias.
Thank you so much, Alejandra. Thank you to Alejandra, Dr. Burgess, and Dr. Kaufman for those fabulous presentations. I know we're getting close to time. We'll ask one question and have a little conversation and then pass it back to the facilitators. Alejandra started to touch on it at the end in terms of private sector engagement. We talked a lot about on-farm measures in the panel. And in many cases, the private sector is playing a huge role and could play a larger role in providing tools and solutions. So I'd love to hear from the panelists what additional efforts you would like to see from the private sector to help meet the challenges of the screwworm spread in Latin America and the US. And I'll pass it to whichever panelist wants to tackle this first.
All right, I'll start and try and keep it brief as we're running out of time here. Our Texas cattle producers and wildlife organizations have been incredibly important in raising the awareness of the situation with our state regulators and legislators, and those individuals carry considerable weight. The other aspect that is important is to continuously have available the updated economic impacts, because that's what drives a lot of this. And, you know, right now there are no cattle imports allowed from Mexico into the United States, and that's an incredibly important economic driver for Mexico. And those border crossings likely will not be reopened until the screwworm is pushed out of Mexico or other assurances are provided. And those sort of messages delivered by the private sector can help the governments leverage the support they need to in order to help us, right? We don't have any control over some of these things, but sharing information with the private sector can in fact help leverage some of those requirements for this.
Bueno, voy a continuar yo. Coincido con Phil. Efectivamente, se requiere una mayor participación del sector privado, pero más que partícipes, diría que deberían ser integrados en estas estrategias, tanto nacionales y regionales. Entonces, nosotros los estamos incorporando, por ejemplo, como formadores. Entonces, entendemos que el sector privado organizado tiene suficiente capacidad pues organizativa y de llegada a sus socios miembros. Y que además de eso, lograr el intercambio entre ellos mismos. Hemos distinguido que es completamente diferente cuando un experto le dice a un productor qué es lo que tiene que hacer, cuando compartimos un productor panameño, un productor costarricense, hablándole al productor mexicano. Entonces, en esos talleres de intercambio realmente es valiosísimo en la serie de mensajes y lecciones que podemos sacar en conjunto. Consideramos integrarlos a ellos, incluso en estos aspectos de toma de decisiones, en comités nacionales, en comités regionales, resulta relevante.
I would add that to get private sector more involved in this, we need to understand kind of what procedures look like in these different Latin American countries. It's not it's not always easily accessible, and involvement with private industry really starts with what what usage patterns and kind of what strategies are being used so that we can come up with ideas for how private sector can leverage their time and resources towards generating new types of technologies, or in the case of chemical control, new types of products that can be used. So that's really an important kind of intersection between the two of them.
Great. Thank you for sharing those calls of action to the private sector around economic impact and procedures and knowledge sharing. With that, unless there are any pressing questions for this panel, I'm going to pass it back to you, Joshua, for the final session.
Thank you very much, Jasmine. I will pass on to Eugenia.
Well, I just want to thank you so much, Jasmine, and the panelists. And I think this is such a great webinar. And I would like to invite all the panelists to turn on the cameras and maybe do a quick, Andrew, if you are, if you agree with me, do a quick closing on what's the main areas that we can identify. This is the first webinar. I'm pretty sure that we are going to keep having more meetings. Maybe not in a webinar, but just retrieving what we learned from today. So I pass to you, Andrew, if you want to say some words, and then we can start the dialogue.
So thank you, Eugenia. I mean, I agree. I think this has been a great kind of just putting a lot of different pieces on the table and seeing different perspectives and where people are, and how we can kind of start integrating all those things together into a new strategy. I also think we need a lot of work in a lot of these areas, and from training to new control measures to communication about the issue, new treatments, more surveillance and risk management. I mean, we need kind of all of these things are necessary, and getting kind of a team approach is really going to be the best way to make progress forward. In the sake of time, that's really just my takeaway. I'm really excited about getting this started, and I'll pass it back to Josh or
Bola.
Thank you very much. Caught up the echo here. Yeah, I echo those comments. I thank you all so much. It's been very interesting. I have personally learned a lot. There is a vast amount of expertise here and complementary skill sets, experience, and so on. So I think there's there's so much to be gained by pooling our resources, pooling our expertise, and working together as a team on this. I think there are a few others that we need to consider bringing in, like those from USDA. We also talked about the industry, and so there maybe we can give some thought to that, and who else would we need to bring in to complement this team. And then, for those of us on the research side, things only happen when there's funding. And so I think we also need to strategize about that. And we are very grateful to the donors we have here on this call. So Jasmine, thank you very much for your participation, Eugenia, and the others from the funding community. But I'm hoping this is just a launch pad for greater things. And I look forward to seeing how this develops. Thank you.
Thank you, Bola, and for sure. I think I have at least a lot of 23 pages of notes and a screen that I took from the presentations. So I'm pretty sure that we can move forward with activities that we can support for Latin America, but at the same time working with the UF and many other institutions that joined today with us. And for sure, this is something that we need to approach together. There is no way that we cannot use everything that was researched before and is still in place under research and not take that. So we cannot start from zero. We have a lot that we have done. I'm very curious actually on understanding, what kind of short actions or we have to take and communicate to farmers, but not only farmers. I also think about the transportation. Because I remember when I was moving cattle in the farm, and you put cattle in the transport, in a big truck, there is something that need to be, we need to do some work with them too, some kind of protocols, and maybe in every step where the cattle moves. So, and then of course, I love what Phil shared with us as short for actions for short-term, medium-term, and long-term in science and in the rest of the partners. So I would like to say that we are going to work with Josh in a summary, probably a technical note and a policy brief that we are going to share with all of you. So you can also add your insights and your ideas. And then as we plan, we are going to move on a second phase of this, which is a in-person meeting at the University of Florida at some point after October, and work from that as well. From FONTAGRO side, thank you so much. I don't know if someone want to say something, or, I want to give a space to all of you just to make a short comment as well. Maybe Andrew or Justin, Lindsay, Daniel, Jasmine, Alejandra. If not, well, thank you so much again. I want to express my gratitude to all of you, to ICA, University of Florida, Texas A&M, INEA Uruguay, and Alejo have to leave early, and Oklahoma State University, and the Foundation for Agricultural, Food and Agricultural Research as well. Well, thank you very much, as it has been a pleasure. Oh, Daniel, you have your hand.
I just wanted to put one more short thing out there. I spoke mostly about sterile insect technique, but I also wanted to say to the group that our laboratory works on artificial intelligence approaches to trapping and surveillance, as well as environmental DNA approaches. And so we are actively seeking collaborators in the native range of this animal where it's endemic, so that we can go down and sample, test these traps that we've been working on, and find folks who would be willing to work with us on that, as well as the SIT sides of our project. So if you have anybody who's really interested in these areas, please ask them to reach out to me. Thank you.
Absolutely. Thank you for sharing that with us. So thank you very much, everyone. And Josh, I pass to you.
Thank you, Kenya. just to say another thank you to all the panelists and moderators who joined us, and thank you to everyone who joined us via YouTube. We're going to end the transmission now, but I would ask all the speakers to please stay on a moment longer so that we can take a lovely photo. We will be uploading the video of the workshop both in English and Spanish on our YouTube channel following the event. And I just wanted to highlight a couple of comments that stood out to me as well. So there have been challenges highlighted around quarantine, dispersal, the role of what the role of the university sector could be in bringing value to these processes. And a comment from Jose Ordaz, who couldn't stay for the meeting, but at the start he mentioned laying out a roadmap, not just for the control of the screwworm, but for the eradication. And I think that would be something great to aspire to.
Thank you, Josh. So the photo is taken, right?
We're going to take it now. So
Okay.
if everyone can smile. There we go. Thank you very much.